So, hello students, how are you all? So, I hope that you are well prepared for your NEET and your practice and revision must be going on. So, this is a very important and crucial time, and that's why we are here with this one short video in which we will cover the entire botany. Now, as the title of the thumbnail suggests that this video is aiming for you to score 180 out of 180. Now, 180 out of 180, yes, it is possible. At least in bio, it is definitely possible. So here, let's first make some rule for this video. The important is you have to be very attentive. Definitely in between, this is going to be a long session. In between, I will give a break of few minutes. But don't randomly go away from the lecture, because ultimately it will be very fast and you will miss some of the point and you will not get the benefit. See, 180 out of 180 is possible and desired in botany and zoology and overall bio also, because in physics, chemistry, things are not that predictable, and you know that in bio every question is going to be from NCERT. So it is easy, it is possible. So let's start with the class. So here I will cover it chapter by chapter and I will cover it from NCERT. So here what you have to do, first thing is that whenever I start a chapter, let's say you have a particular doubt in a particular topic, just in the comment you write the name of the topic. I will get some idea that what are the main topic in which you are having more problem. And that's why I will explain those topics in a little detail. Don't worry, I will cover each and every point, and that's why I'm sure that this will help you to get 180 out of 180. So let's start with the video. So if all of you can hear me clearly and all of you have joined it, so give me a thumbs up. So let's start with the video. So here in this botany, the first chapter is the living world. Now, this is one of the smallest chapter. This is one of the smallest chapter, and from here, usually one question come in NEET. Usually one question, or sometime even not a question. So this is comparatively a less important chapter, but yes, even if one question is asked from here, still you have to cover it properly. So here in this chapter, living world, the main point from where the question is asked, so as you can see here in this case, number of species is given. So number of species, you have to remember that total number of species is 1.7 to 1.88 million, and this constitute the biodiversity. Biodiversity is all the plants and animal present on the earth, all that constitute biodiversity. Now here another important point is the binomial nomenclature. So from here question is asked, what are the rules of binomial nomenclature? So binomial nomenclature will include two words. First one will be the genus, another one will be the species. Second rule, genus is written with capital letter, species is written with a small letter. Both are separately underlined or written in italics. They are derived from Latin word. These are the important point. Fourth point, the name of the author can also be included, and name of the author is written as the third letter in small. For example, Mangifera indica, Lin. Lin means Linus. Linus has discovered it, so it is Lin. Now another point from here is there are few terms like taxonomical categories. Taxonomical categories. Another term usually used here is taxon or taxa. Another is taxonomical rank. Taxonomical rank. They all are unit of classification. Remember, all of them have the same meaning. All of them has the same meaning. So there are total 7 categories. So taxonomical categories They are the unit of classification. You call them rank, category or taxon. Species, there are 7 categories as shown here. These are the 7 categories. The lowest category is species and the highest category is the kingdom. So there are total 1, 2, 3, 4, 5, 6, 7, right? King Philip cut open 5 green snake, right? So this is 7. So this is called the taxonomical hierarchy. Simple, from this hierarchy question is asked. Then here what you need to understand, there are several species given. For example, Mangifera indica, Solanum tuberosum, Solanum melongena, Solanum nigrum. So you have to understand Solanum is the common genus and it has different species. Similarly, in case of genus, if we say Panthera, so Panthera is a genus in which there are several species. Panthera tigris, Panthera leo, Panthera paradus. So from here question can be asked, what is the genus of this particular animal? What is the binomial nomenclature of this particular animal? So that you can easily answer. Similarly, family. So family of cat is called Felidae. Family of dog is called Canidae. And they both come together in the under which order? So they both comes under the order of Carnivora. Similarly, phylum, kingdom. So these all are very basic points which you can remember. Then this table is also important. Here you have to remember the family, order, class of all the organism. So these are the basic point. If question is asked, question will be from here only. Nothing much is there in this chapter. Second chapter, biological classification. It is a very important chapter because approximately 3 to 4 question is asked from here. 3 to 4 question is too much, right? So this is an important chapter. So here in this case, first of all, it was Aristotle who first of all talked about the classification, and he classified animal also and he classified plant also. He classified animal on the basis of whether it is red blood or without red blood. Red blood or without red blood, he divided the animal. And in plant, he has divided them into the plants, he have divided into trees, herbs and shrubs. So these were all based on some gross morphological character, some rough morphological character. They were not sufficient. But yes, if question is asked, who first of all did the first scientific classification? So it was Aristotle. Later on, Linus has given the two kingdom classification. The two kingdom classification was given by Linus, and he classified organism on the basis of presence or absence of cell wall. Presence or absence of cell wall. Now, when we say cell wall present, then it is plant, cell wall absent, then it is animal. But this classification also have several limitations, and limitations were prokaryote, eukaryote kept together, then the multicellular, unicellular kept together, autotrophic, heterotrophic kept together. So these were certain limitations. Position of certain organism like Euglena was not very much clear. So due to this, two kingdom classification was not sufficient. So what we get? We get the 5 kingdom classification. In the 5 kingdom classification, there were several other factors which were involved, and these factors included the cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationship. So all the drawbacks which were there in the 2 kingdom classification were withdrawn or were addressed in the 5 kingdom classification. Here this table is very important. From this table question is usually asked that how many of these kingdom are prokaryotic, how many are eukaryotic, how many are without cell wall, how many are with cell wall, how many is heterotrophic, how many is autotrophic, which kingdom has cell wall made up of chitin, Which kingdom has cell wall made up of polysaccharide and amino acid? So in this way question will be asked. Which kingdom has holozoic nutrition? Which kingdom has both autotrophic and heterotrophic nutrition? So many kind of question is formed from this. Then there are few examples given. For example, Chlamydomonas and Spirogyra are pair of unicellular and multicellular, which is the drawback of two kingdom classification. Similarly, here given Chlamydomonas and Chlorella, which are autotrophic, are kept together with Paramecium and Amoeba, which is heterotrophic. Again a contrast. So these are some of the examples given. Then if talk about the first kingdom, that is Monera. So Monera are the kingdom of prokaryotes, unicellular prokaryotes. So Monera are unicellular prokaryotes and they are of four different shapes. It is Bacillus, Coccus, Spirilla and Vibrio. So these are the four shapes in which this bacteria or Monera is present. Further, they have, this is an important line, they are simple in structure but complex in behavior. They can be both autotrophic, heterotrophic. They can be photoautotrophic also, they can be chemoautotrophic also. So here see, they can be chemosynthetic autotroph or they can be photosynthetic autotroph. Depends what? They are chemosynthetic if they use energy from inorganic substrate, and if they use energy from organic substrate, then it is if they use energy from the light, then it is then it will be called as the photosynthetic autotroph. So autotroph is of two types, photosynthetic and chemiosmotic or chemisynthetic. Now autotroph and heterotroph. Most of the bacteria is heterotroph. Most of the bacteria is heterotroph. Very less number of bacteria is autotroph. Autotroph can be chemiautotroph and photoautotroph. Then this bacteria is divided into archaebacteria, eubacteria. So let's see them. Now archaebacteria, they are extremophile, find in harsh habitat, extreme condition. They can be halophiles, they can be thermoacidophiles or they can be methanogens. Halophiles if they are found in high salt concentration. Thermoacidophile if they are found in high temperature or high acidity. Methanogens, they are found in marshy areas. What are the character of archaebacteria which make them survive in a tough condition? So they have a cell wall structure which help them to survive. Now methanogens are found in three locations. First one, they are found in marshy areas, submerged paddy field. Second, they are found in rumen of intestine of ruminant animal like cattle. Third is they are found in biogas. Further, if we continue, then eubacteria is also called the true bacteria. Eubacteria is the true bacteria. Eubacteria can be photosynthetic, that is cyanobacteria. Cyanobacteria are considered as photosynthetic autotroph. Now these cyanobacteria can be unicellular, it can be filamentous or it can be colonial. This is a filamentous bacteria which is nostoc. Nostoc, here you can see labeling is important. Mucilaginous sheath is there, heterocyst is there. Heterocyst is responsible for nitrogen fixation. So if question comes, can cyanobacteria do nitrogen fixation? Yes. Can bacteria do can cyanobacteria do photosynthesis? Yes. So this is the characteristic of cyanobacteria. So remember heterocyst. Now bacteria can be chemosynthetic autotroph also. Chemosynthetic autotroph will use nitrite, nitrate as the inorganic substrate. Nitrite, nitrate and ammonia. And here the example will be purple sulfur bacteria, green sulfur bacteria, sulfur bacteria. But remember here the chemosynthetic autotroph will not release oxygen. They will do Anoxygenic photosynthesis. Anoxygenic photosynthesis, right? Now, heterotrophic bacteria can be of several types, right? So they can be performing, they can convert the milk into curd, lactobacillus, right? Then they can cause certain disease, so they are pathogenic also. Bacterial disease, you remember the name of some bacterial disease like cholera, typhoid, tetanus, citrus canker, these all are bacterial disease. Further, bacteria reproduce by fission. Then bacteria in unfavorable condition can form spore, or also called as endospore. So spore formation is the characteristic of bacteria in unfavorable condition. Then another form of bacteria is mycoplasma. Mycoplasma is the only bacteria without cell wall. If question comes, which prokaryote is present without cell wall? So the answer is mycoplasma. Now, can mycoplasma survive without oxygen? Yes. Is mycoplasma pathogenic? Yes, they are pathogenic to animal and plant. So this is all about Monera. Now, Monera are considered, or bacteria are considered to be simple in structure and complex in behavior. Complex in behavior because they show metabolic diversity. They show metabolic diversity. That is why. Now coming to the next kingdom. So the first kingdom completed. Second kingdom is Protista. So Protista it is a kingdom of unicellular and eukaryotic organism. Protista will include unicellular and eukaryotic organism. In Protista, it is of different types. So first type is the chrysophytes. Chrysophytes are also called as diatoms, also called as golden algae or desmids, which are found in fresh water and they can float passively. Now here chrysophytes have several characters. For example, they have cell wall made up of cellulose. Now cellulose has silica deposition, right? So due to presence of silica deposition, they are referred as diatomaceous earth. Due to presence of silica deposition, they will accumulate and they are indestructible. This, due to presence of silica, the diatoms can be used for polishing and filtration and filtration of oil and syrup. Diatoms, they are photosynthetic, so they are the chief producer of the ocean. See, in such case, you have to keep a bullet points that how many points are there in chrysophytes, and simply this side you keep on writing it, that they are also called as found in, then passive floater, then they have silica in cell wall. Further, this silica make them non-destructible. Further, due to presence of silica, they have this much of use. In this way, each topic you can simply cover and keep on revising again and again. Then further, dinoflagellates. So as the name suggests, dinoflagellates means two flagella is there. Two flagella. So two flagella, longitudinal, and so it has two flagella, and one of the flagella is longitudinal, another is transverse. As you can see, it has two flagella, longitudinal and transverse. Here cell wall is also made up of cellulose, but here cell wall is present in the form of cell plate. Now if question is asked, cell plate is a characteristic of, so you will say dinoflagellates. Further, if question is asked who is responsible for red tide, you will say dinoflagellate. Which dinoflagellate is responsible? So it is Gonyiolex. Now they are photosynthetic or not? Yes, they are photosynthetic and they release toxin which can kill other aquatic animal also. Again here few points are there, you just list it down, move ahead. Euglena, again Euglena has two flagella, the small and the large one. They are also found in fresh water, but Euglena has one character, that they lack cell wall. They lack cell wall, instead they have pellicle present. Pellicle is the protein layer present outside the euglena. Euglena have a unique mode of nutrition that is called mixotropic nutrition. Mixotropic means in the presence of light they are autotropic, in absence of light they are heterotropic in nature. That's why in two kingdom classification, during the two kingdom classification, the position of euglena was not known properly, not defined properly, because it has no cell wall and at the same time it was showing characteristic like the plant. Slime mold. So slime mold has a character similar to fungi because it is heterotropic and also it is saprophytic mode of nutrition. These slime molds are saprophytic. In favorable condition, they form a mat-like structure, and that mat-like structure, this is the mat-like structure, and this is called plasmodium. This plasmodium is different from what we see in protozoa. So they form mat-like structure called plasmodium, but when the condition is unfavorable, they will form a fruiting body. They form a fruiting body, and fruiting body has spore, and these spore have true walls. The spores have true walls. Then further, protozoans. So protozoans are divided on the basis of locomotory organ. They are divided on the basis of locomotory organ. So it is called amoeboid protozoan if the locomotory organ is pseudopodia, also called as false foot or pseudopodia, that is amoeboid. Example of amoeboid are amoeba and entamoeba. Another one is the flagellated protozoans. Flagellated protozoans will have flagella as the locomotory organ. Example is trypanosoma. Flagella as the locomotory organ, example is trypanosoma. Ciliated protozoa, cilia is present as the locomotory organ, and the example is paramecium. Sporozoans, they have no locomotory organ, and they are the deadliest pathogen. Malaria parasite, that is plasmodium. So this is all about the Protista. So if we summarize here Protista, we find that Protista is unicellular eukaryote, but they have organism which has cell wall also. Some are without cell wall also. Some are having plant-like character. Some are having fungi-like character, and some are having animal-like character. Animal-like character, protozoa. Plant-like character, dinoflagellates and diatom. Fungi-like character, slime mold. Then the third kingdom is kingdom fungi. So kingdom fungi, it is saprophytic, that is heterotropic mode of nutrition, and it is saprophytic in nature. This saprophytic in nature, so it feed on the dead and decaying matter. So they can be parasitic also. Parasitic include rust and smut. They are rust and smut, which will be parasitic. If their body type I say, so body type is filamentous, which is called hyphae. Filamentous, it is called hyphae, and when there is a network of hyphae like this is called mycelium. Network of hyphae is called mycelium. Now this mycelium can be of two types. They can be septate, means septa is present and each cell having one nucleus, or they can be aseptate. Aseptate means the septa is absent. When it is aseptate, then it is called cenocytic, right? So they can be cenocytic, aseptate, or they can be septate. Since they are filamentous, so they will divide through fragmentation. But Apart from that, fragmentation, budding is also there and fission is also there. So this is about the vegetative propagation. So vegetative propagation through fragmentation, fission and budding. Now what about asexual reproduction? So asexual reproduction take place through the formation of asexual spore, and these asexual spore can be conidia, sporangiospore or zoospore. Now here remember, apart from this there is one more spore which is aplanospore. Actually aplanospore is a type of sporangiospore only. Sporangiospore is divided into two parts, zoospore and aplanospore. Zoospore is motile, aplanospore is non-motile in nature. So all of these are the asexual spores. So remember, question is asked from here, what are the asexual spores? Then if we talk about the sexual spores, so sexual spore include oospore, ascospore and basidiospore. So remember the sexual spore also. Question is asked, which of the following is sexual spore, which of the following is asexual spore? Then further, the sexual reproduction will include plasmogamy, karyogamy and then the meiosis in this zygote. So sexual reproduction represent three steps. The first step is plasmogamy, in which two cells, the protoplasm will fuse. In karyogamy, two nucleus will fuse, and finally zygote will undergo meiosis. But it is not always in this case. In some case, the karyogamy is not immediately followed by the plasmogamy is not immediately followed by karyogamy, and this give rise to a stage called dikaryon stage. Dikaryon stage. So what are the organism in which this dikaryon stage is found? So dikaryon stage is found in ascomycetes and basidomycetes. Remember, ascomycetes and basidomycetes. In these two cases, the dikaryon is present. Dikaryon is present. So where dikaryon is absent? So dikaryon is absent in the first one, that is phycomycetes. So now we will see in how many classes the fungi is divided. These are some of the general character in which we have seen the body type, vegetative propagation, asexual reproduction, sexual reproduction. Now individually we will see different class of fungi. So the first is phycomycetes. So phycomycetes are basically, these divisions in fungi is actually made on the basis of their mycelium, mode of nutrition and the storage product. So here let's see here in case of phycomycetes, first of all the mycelium is here, the mycelium is aseptate. So it is aseptate and cenocytic, point number one. Point number two, reproduction. So first is vegetative propagation. So it will be through the fragmentation. Then comes the the next case, if it is about the asexual reproduction. So asexual reproduction through the formation of zoospore or by aplanospore. Zoospore or aplanospore. But sexual spore is by the formation of, it is by formation of zygospore. So remember, in phycomycetes there is asexual spore and there is sexual spore. Asexual spore is zoospore, aplanospore. Sexual spore is zygospore. These sexual spore or gametes, these gametes can fuse through isogamy, anisogamy and oogamy. Three example you remember, Rhizopus, Albugo and Mucor, out of which one is bread mold? Rhizopus is the bread mold. So how question will be asked? First, phycomycetes, they are there is no dikaryon stage, asexual spore, sexual spore, their example. Then the next one is ascomycetes. Ascomycetes is also called sac fungi because they form a sac which is called ascus. Sac is called ascus. Now here in this case, this ascomycetes, one of the important part is the Penicillium notatum, from where the antibiotic penicillin was obtained. Further, here the mycelium will be septate and it will be branched, septate and branched. Then here the asexual spore will be conidia, sexual spore will be ascospore. Now remember here, very important, conidia is exogenous, whereas ascospore is endogenous. So can we say that in case of ascomycetes, the asexual spore formed is exogenous in nature and the sexual spore formed is endogenous in nature? Here, if I further draw, let's say this is the ascocarp, inside is the ascus, and inside the ascus is the ascospore present. As you can see, this is ascocarp, ascus and ascospore. Clear? So these are then example. Very important here in all the cases the example. Aspergillus, claviceps, neurospora. Neurospora is also used in genetic work. Further, morels and truffles. Already we have seen penicillium. So penicillium is also example of this. Further, basidomycetes. So basidomycetes, very important. Basidomycetes is also known as the club fungi. Here they include bracket fungi, puffball. They can also be parasitic in nature. Rust and smut fungi are actually part of the basidomycetes. Mushroom is also comes under basidomycetes. Here the mycelium is septate. See, except the phycomycetes, all will be septate and branched. Now here asexual spore here will be absent. Sexual spore will be the basidospore. This basidospore is what? Exogenous or endogenous? So it is exogenous. So now here what we can say, this will have asexual spore absent and the sexual spore present in the form of basidiospore. Further, here if we see the example, so example include agaricus, eustylago and puccinia, which is the red fungi. Here the example is agaricus, eustylago, puccinia. These are the example. Further, already seen rust and smut fungi, bracket fungi, puffball, all are example. Now which is the smut fungi? Eustylago. Which is the rust fungi? The puccinia is a rust fungi. Then deuteromycetes, also called as fungi imperfecti. Why is it so? Because they have sexual reproduction absent. So the sexual phase is absent. So now, is asexual spore formation is present? Yes, asexual spore is formed in the form of conidia. Vegetative propagation present? Yes, it is in the form of fragmentation. So in deuteromycetes, the asexual spore is absent, that's why it is called fungi imperfecti. Deuteromycetes is also called as the form group. Here, if the sexual stage is discovered, then it will be transferred into either into the ascomycetes or basidomycetes. So that's why it is also called a form group. Here the example is Here 3 example: Alternaria, Colatotrichum and Trichoderma. Alternaria, Colatotrichum and Trichoderma. These are the 3 examples. Now further, so I think here Kingdom Plantae, Animalia, they are covered separately. So we will leave that part. Now let's see the other part, which is the virus, viroid, prions and lichens. So first of all, this diagram is important. You should be able to label these diagrams. You can be given the diagram A, B, C, D, you have to label. So that is important. Then here name of 3 scientists in virus is important. The first name is Dmitry Ivanovsky, second name is Byjernik and third name is Stanley. What are their contribution? So Ivanovsky has discovered the virus. Byjernik has called virus as the Contagium vivum fluidum and Stanley has crystallized the virus for the first time. So these are the contribution of these 3 scientists. Virus is actually a nucleoprotein, means virus will have protein and it will have nucleic acid. Now this nucleic acid can be DNA, nucleic acid can be RNA. Protein are called capsid. It has the unit which is capsomere. Unit is capsomere. Is it clear? So remember, this nucleic acid can be DNA or RNA, but it can never be both. No virus contain both RNA and DNA. Now there are certain names given. For example, in case of animals, the animal virus, in case of animal virus, the genetic material can be double-stranded DNA, it can be single-stranded RNA, means it can be both RNA and DNA, whereas in plants it's mostly single-stranded RNA. Now here another example is also given for bacteriophage. Now bacteriophage, mostly they have double-stranded DNA as the genetic material, but there are variation present, right? Bacteriophage QB, it has single-stranded RNA virus, single-stranded RNA as the genetic material. So here these points are given. Then some disease are given. What are the viral disease? Mumps, smallpox, herpes, influenza, AIDS. These are some of the viral disease. These viral infection in plant cause certain symptoms like leaf rolling, curling, yellowing, vein clearing, dwarfing and stunting of growth. So these are some of the points about the virus. Further, viroid discovered by Theo Diner. It causes which disease? Potato spindle tuber disease. This viroid is smaller than virus. It is smaller than virus and it has low molecular weight RNA. Low molecular weight RNA. It do not have DNA, it do not have protein. So viroid do not have protein and they are infectious because they can cause disease. Disease caused is potato spindle disease. Prions, they are misfolded protein or abnormally folded protein. They can cause what kind of disease, what category of disease? So they cause neurological disease. Here prions have a size which is similar to virus, and these agents, prions, cause BSE, means bovine spongiform encephalopathy. In which organism? In the cattle, which is also called the mad cow disease. Remember, this is a neurological disease, so it will cause some abnormal behavior in the organism. Mad cow disease. Similar disease in humans are called CJD, Creutzfeldt-Jakob disease. So this is about the prions. Then further lichens. So lichen, they are the symbiotic association. They are a symbiotic association between the algae and fungi. Here remember, algae act as phycomycetes and fungi act as mycobiont, and algae act as phycobiont, not phycomycetes. I just said phycobiont and mycobiont. Now what is the role of mycobiont? So mycobiont is the fungal component. So fungi will not prepare food. Who will prepare food? The algae will prepare food. So phycobiont will have a role in preparation of food, and mycobiont will have a role in providing shelter and absorb mineral nutrition. These lichens are very important. They are a good pollution indicator because they are not found in the polluted areas. So these are all the points related to biological classification. So it's a big chapter. 3-4 questions are there. So just go through each and every point. See here what is important. What is important? Okay, some students are asking second year. Yes, every part, second year also will be covered. First year, second year, all will be covered. Right. So here see, some students are asking about PYQ. So here we are not covering PYQ chapter wise. What we will do, that for PYQ we will have a separate video in which all the 2017 onward, all the PYQ videos we will cover it separately chapter by chapter. Here let's mainly highlight the all important topics of each chapter. Okay, so let's continue with the next chapter. So the third chapter is plant kingdom. Now plant kingdom, again very important chapter, but at the same time there is a lot of information. So students tend to find this chapter also very not difficult, but it has a lot of examples. So memorization is a little tough here. Also one point is, so all of you who are here connected, please write down which part of plant kingdom you find most difficult. Plant will include algae, it will include bryophyte, pteridophyte, gymnosperm and angiosperm. So angiosperm anyway we will study in the sexual reproduction. So out of this, which one you find it difficult, which part you want me to cover in a little detail? So let me know. So in plant kingdom, basically it starts with the classification, right? So here it says about 3 types of classification. The first one is artificial classification, second one is natural classification, and the third one is phylogenetic classification. Phylogenetic classification. Artificial classification, here a large number of character is used, right? All possible characters. In artificial only few characters are used. Large number in natural classification will be used. See, artificial, it will have only few character, and that also the external character, that is the morphological character. The artificial classification was given by Linus. It was given by Linus. Natural classification. Now, in artificial classification, one drawback was, it is given, one drawback of artificial classification was it considered vegetative and reproductive character equal. That was not in case of natural classification. It was based on natural affinity. It included external as well as internal character, and here the sexual character given priority over vegetative character, right? This natural classification was given by Bentham and Hooker. Polygenic inheritance, here the evolutionary relationship is considered, or phylogenetic relationship is considered, then it is phylogenetic classification. Similarly, there are 3 types of taxonomy given: numerical taxonomy, cytotaxonomy and chemotaxonomy. Numerical taxonomy, all character will be used. So here computers are required and programming is required. If the cellular character is considered, cytotaxonomy. If the biochemical character is considered, it is called chemotaxonomy. Then further, next let's enter into the part. So yes, I can see many of you have written the points. Okay, life cycle. Sudhakar is asking for life cycle of bryophyte, pteridophyte and their life cycle. Yes. So I know that this is one of the most common doubt of all student here. Okay. So I will cover that. So let's start with algae. So algae, very simple, they are photosynthetic in nature, right? Now algae, these are some of the algae. Algae is divided mainly into three parts. The green algae, that is chlorophyceae, then brown algae and then red algae, rhodophyceae. So here in this table it is explained very nicely. So the green algae has which pigment? Chlorophyll A and B. Chlorophyll, so their distinction is on the basis of pigment, stored food, cell wall, flagella, and also they have different habitat. So first if we see the pigment, then green algae has chlorophyll A and B, brown algae has chlorophyll A and C, and red algae has chlorophyll A and D. So this is the basis of pigment. Why brown algae has brown color? Because of the pigment fucoxanthin. Red algae is red because of phycoerythrin or R-phycoerythrin. Which is the stored food in green algae? So it is starch is the stored food in green algae. Stored food in brown algae is mannitol and laminarine, and in red algae it is Floridian starch. Cell wall all have cellulose as a component, but there is slight difference. In case of green algae it is cellulose, right, and some other are also lactan, mannan, you will see it in cell, lactan, mannan and calcium carbonate is also present. Now here this cellulose and algine. Algine is a part of cell wall of brown algae. Algine is also a hydrocolloid. Then Floridian starch, rhodophyceae. Rhodophyceae has a storage food of Floridian starch. Then the cellulose, pectin and polysulfate ester. So these are the composition of cell wall. Then flagella. If we see green algae, it has 2 to 8 flagella. If we say brown algae, it has 2 unequal flagella, which is lateral, and in red algae no flagella is present. Remember, no flagella present in red algae, right? Then non-motile stage, red algae. Pyriform spores or gamete are present in, so pyriform in brown algae. Agar is obtained from red algae. Which red algae? So Gelidium and Gracilaria. Algine is obtained from brown algae. Carrageen is obtained from red algae. Algine, carrageen and agar, they all three are considered as the is considered as hydrocolloid. So this is simple, you just need to read it. Here examples are very important, right? Examples you should need, and these diagrams. Question is usually asked, you will be given this and you will be asked, this is volvox, right? Then this one, Eulothrix. Then this one, Laminaria. Then fucus. You should be able to identify. Fucus has air bladder in it. Then Dictyota. Now which one is the largest one? So brown algae is the largest one. They are large, so the body is divided into holdfast, then stripe and frond. Frond is photosynthetic in nature. Then the red algae, Porphyra and Polysiphonia. This is Porphyra, this is Polysiphonia. So diagram is important, example is important here, and character of course is important, which I have just shown. So then comes the important part, that is bryophytes. Now in bryophyte, if in general if I talk about the structure, it is more or less clear, but here more important is its life cycle. So here I will take some time to explain the life cycle. All of you can note this life cycle down. So I am taking a separate page to explain the life cycle. See, first of all, when I say Bryophytes. So bryophytes have the main plant body made up of, so the main plant body is gametophyte. Main plant body is gametophyte. It will have two sex organ, antheridia and archegonia. Antheridia and archegonia. These sex organ are multicellular. Sex organ are multicellular. This male organ, this is female organ. Female organ is flask shape. Female organ is flask shape. This one, antheridia, become antherozoids. Means these are the male gamete. Now male gamete is biflagellated. Biflagellated. Female gamete, this is egg. Now this both will fuse in water. Now because they fuse in water, that's why they are called amphibian of plant kingdom, because fertilization they need water. So this fusion will form zygote. Fusion will form zygote. This zygote will develop into sporophyte. See, remember, line is given, zygote do not immediately undergo meiosis. Zygote get converted into sporophyte. This sporophyte is multicellular. Sporophyte is multicellular. So it has three part. It has three part, which is foot, seta and capsule. Foot, seta and capsule. Three parts are there. Now out of these, capsule has a special cell which is called spore mother cell. These spore mother cell undergo meiosis and form spore. Now these spore can directly form gametophyte, or they can indirectly form gametophyte by forming protonema. Then secondary protonema, and then they form the gametophyte, right? So this is overall the life cycle. Once again I will repeat, the main plant body is gametophyte, gametophyte is haploid, sporophyte is diploid. This gametophyte will have two sex organ, one is antheridia and archegonia. Antheridia is male organ, archegonia is female organ. These sex organ are multicellular in nature. Antheridia will form male gamete, antherozoid, archegonia form egg. These both fuse together in water to form zygote. Zygote will be 2n. Sporophyte will be 2n. Foot, seta, capsule, all of them will be 2n in nature. Meiosis will produce spore. Spore will be n because meiosis has happened. Now here if I divide, then this from here onward, right? From the zygote everything is diploid, and above that everything is haploid. So haploid is called gametophytic generation, diploid is called sporophytic generation. Now here if I say sporophyte is non-photosynthetic and gametophyte is photosynthetic. So always non-photosynthetic depend on photosynthetic. So here in bryophyte we say that the sporophyte is dependent on gametophyte. Sporophyte is dependent on gametophyte. Here remember, spore form the gametophyte in case of liverworts, and here in mosses. In mosses they don't directly form gametophyte. In mosses they first form protonema, then they form secondary protonema, and then the gametophyte. Here remember, protonema is filamentous. Protonema is filamentous and secondary protonema is leafy stage. Secondary protonema is leafy in nature. So this is overall the life cycle of bryophytes. Is it clear? Now, one important point is difference between liverworts and mosses. So difference lies in thallus. In case of liverwort, thallus is dorsiventral. In case of mosses, thallus is prostrate or erect. Second difference is in rhizoid, unbranched, unicellular in liverwort, branched and multicellular in mosses. Another difference is the sporophyte. Sporophyte is more elaborate in mosses, less elaborate in liverwort. Then another difference, the spore dispersal mechanism. Spore dispersal mechanism is more elaborate in mosses, less elaborate in liverworts. Then further, if you see, mosses form protonema, liverworts will form gemma cup. As you can see in the diagram, they form gemma cup. So these are some basic difference between liverworts and mosses. So those all you can read and you can figure it out. Mainly this life cycle is important. So I guess that you all have understood the life cycle. So if you understood, give me a thumbs up. Okay, great. So let's continue with the topic. So now if we see further here, so this is about the bryophytes. Now if we see, mosses are given here, mosses. So the difference between mosses and bryophyte, if you read it, you will get it, which I have just summarized. Example you have to remember: Funaria, then Polytrichum, Sphagnum, they are the example of mosses. Liverworts, example. So here diagram, just go through it once. If you see the diagram here, as you can see here, Mercantia, right? Mercantia. Bryophytes are homosporous. Now many times students get confused that, sir, this is a female thallus and this is male thallus, then why we are calling it homosporous? Isn't it heterosporous? So the answer is no. This homosporous and heterosporous has nothing to do with separate female and male thallus, right? All the bryophytes are homosporous. All bryophytes are homosporous. Just by seeing the diagram you can identify all the organism. Now moving ahead, pteridophytes. So in the bryophytes, the main plant body is thallus, means they are not differentiated into root, stem and leaf. But in case of pteridophyte, it is differentiated into root, stem and leaf. So pteridophyte is the first plant in which true root, leaf and stem has arisen. Also they are the first vascular plant because vascular bundle developed for the first time. Now here these, since they have the root, stem and leaf present, but still leaf is the dominant part of the plant. Leaf is found of three types. The first leaf is small leaf called microleaf or microphyll. There is large leaf called megaphyll, given in NCERT. And third is reproductive leaf, which is called sporophyll. So the leaf can be of these three types. Now in life cycle of pteridophyte, these microphyll and megaphyll don't have much role because they are the vegetative part. Sporophyll has a very important role in the life cycle of pteridophyte. So let's see the life cycle of pteridophyte here. So in case of pteridophyte, the main plant body here will be sporophyte. Remember, main plant body here is a sporophyte, which is 2n. Now this sporophyte, the sporophyll represent the sporophyte. Basically they have the reproductive organ called cone. Cone is also called strobili. Now cone, how does it look like? So cone looks something like this. And here is the sporophyll. Individual sporophyll is present. So this leafy structure with sporangia is called sporophyll. So this is a cone with 1, 2, 3, 4, 5, 6 sporophyll. So cone consists of sporophyll. Several sporophyll form cone. So if I have to draw the diagram of sporophyll, this is sporophyll, single sporophyll with sporangia. What is this structure? Sporangia. So sporophyll has sporangia. This sporangia has, inside this sporangia is the spore mother cell. These spore mother cell will undergo meiosis, and these meiosis will produce spore. Now this spore will be haploid. Before this all are diploid. After this you will get haploid. Then these are the spore. This spore will develop into gametophyte. Now these gametophyte are called prothallus. Now if you read NCERT, it is given gametophyte is photosynthetic, multicellular, right? And they have thallus body. So we say that pteridophyte has true root, leaf, stem. This we talk about the sporophyte, not about the gametophyte. Gametophyte do not have true root, stem and leaf. This gametophyte further will form two sex organ, antheridia and archegonia. Antheridia and archegonia. Antheridia will form male gamete, that is antherozoids, and this will form the female gamete, that is egg. Now here both will again fuse together. Now here also water is required, although they are not called amphibian, but still water is required. But here the requirement of water is comparatively less, means moisture is just required. So then they will form zygote. Zygote will develop into embryo. Embryo will develop into sporophyte. So this is overall the life cycle of pteridophyte. Now if we see from here to here, this is the gametophytic generation, this is the sporophytic generation. This gametophytic generation, the prothallus is photosynthetic. It is multicellular, it is green, it is photosynthetic, as well as sporophyte is also photosynthetic. When both are photosynthetic, when both of them are photosynthetic, then will there be any dependency between gametophyte and sporophyte? No. So that's why in pteridophyte, the gametophyte and sporophyte are independent of each other. They are independent of each other. So this is overall the life cycle. Now here remember, pteridophyte are mostly homosporous. Pteridophytes are mostly homosporous, but few of them are heterosporous. What are the two examples which are heterosporous? So it is Salvinia. It is Salvinia and Selaginella, which is two different, right? Selaginella and Salvinia are two heterosporous pteridophyte. Now when they are heterosporous, so they will form two spore, megaspore, microspore. Megaspore will form female gametophyte, microspore will form male gametophyte. Now when this male gametophyte form gamete, this gamete will enter inside the female gametophyte. When it enter inside female gametophyte, this is called seed habit. This is called seed habit. Remember, there is one line, pteridophyte is the precursor of seed habitat or seed habit. Why is it so? Because here two separate male and female gametophyte are present and zygote is formed inside the female gametophyte for the first time. but that could not develop into embryo, and that is why the seed formation failed. So pteridophyte, no seed is formed. In case of pteridophyte, there is no seed. No seed formation. Although they are vascular plant, for the first time root, stem, leaf appeared, but still they do not have seed. Now here diagram is there. You need to understand, you need to go through it, because question you can be getting the diagram and you have to identify the plants. Then here last part, which is the example, you have to remember that they have 4 class: Coelopsida, Lycopsida, Sphenopsida and Pteropsida. So all the examples you have to remember. Then comes the Gymnosperm. Now Gymnosperm is comparatively more advanced than, Pteridophyte. So Gymnosperm, if we see here, again the plant body will be divided into root, stem and leaf. It is divided into root, stem and leaf. Now in this case also there is no flower, so there will be cone present. Now for root, if I say, root has 2 types of symbiotic association. One is called mycorrhiza, another is called coralloid root. Then stem can be branched or unbranched. Branched in case of pinus, unbranched in case of cycas. Then leaf can also have some xerophytic modification. Leaf can have thick cuticle, they have sunken stomata, they have needle shaped leaf. All this help it in surviving the less water condition. So let's see the life cycle, which is important. So here in Gymnosperm, if we see, in case of Gymnosperm, the main plant body here is also sporophyte. Now here sporophyte is 2n in nature. Here the reproductive organ is cone. Now they all are heterosporous. Remember, bryophytes are all homosporous. Pteridophyte are mostly homosporous, few are heterosporous. Whereas in case of Gymnosperm, they all are heterosporous. When they all are heterosporous, so they will form 2 types of cone. One will be called male cone, another will be called female cone. Male cone and female cone. Male cone will have megasporophyll. Female cone will have microsporophyll. Sorry, reverse. Male will be microsporophyll and female will be megasporophyll. Microsporophyll and megasporophyll. Then megasporophyll will form. Mega will have megasporangia. Megasporangia will have megaspore mother cell. Megaspore mother cell undergo meiosis and form 4 megaspore. Now out of these 4 megaspore, only 1 megaspore will survive, 3 will disappear, and these megaspore will form female gametophyte. This female gametophyte is multicellular. Female gametophyte is multicellular. It has 2 or more archegonia. It has 2 or more archegonia. Now this female gametophyte, this side, now here again we will see microsporangia. It will have microspore mother cell. Then it will have microspore. This will also form 4 microspore, but it will not degenerate, and this microspore will develop into male gametophyte. This male gametophyte is called pollen grain. This pollen grain, now here male gametophyte, female gametophyte, pollen grain. So here pollen grain will enter into male gametophyte. This process is called pollination, and this pollination will be through air. So air is the only mode for pollination in gymnosperm. It enters inside the female gametophyte. Near the mouth of archegonia, the pollen grain is released. Here pollen grain will form the pollen tube. Here it will form pollen tube. So remember, pollen tube formation is there in, pollen tube is formed in case of gymnosperm, pollen tube is also formed. Then inside the female gametophyte, zygote will form. This zygote will develop into embryo, and this embryo will develop into seed, and that will give rise to sporophyte. So this is overall the life cycle of gymnosperm. Here important is that female gametophyte is multicellular. This megasporangia is also called ovule. This ovule has nucellus cell surrounded by envelope. It has nucellus cell surrounded by envelope. So this is all about the sporophyte. Then so this is all about the gymnosperm life cycle. I hope that all of you understood this point. Yes, if you understood it, give me a thumbs up. Okay, so let's continue. So now gymnosperm, here you have to remember this point, very important, that unlike bryophytes and pteridophyte, gymnosperm, the male and female gametophyte do not have independent existence. Now, what do you mean by independent existence? See, here till now what we have seen, till the pteridophytes, we have seen that the male organ is releasing the male gamete, female organ is releasing the egg, and they both are fertilizing in water. Means there is no movement of male gamete inside the female gametophyte. So this movement inside the female gametophyte means dependent, and when there is no movement, then it is called independent. So remember, in case of bryophyte and pteridophyte, it is independent, and in case of gymnosperm and in case of angiosperm, it is dependent. Clear? So this is all about the gymnosperm. Now remember the example: Cycas, Pinus and Zincgo. All three are example of gymnosperm. Angiosperm, we will skip it because anyway we will cover this part in the sexual reproduction of flowering plant, and that is why this part in new NCERT is reduced significantly. Then comes the morphology. So again, morphology is a simple chapter, but here a lot of things you have to remember. For example, in this case, remembering the example in all the topic is a little challenge. So that example, for that you take a sheet of paper, write down all the examples separately and revise it after every 1 or 2 days, then you will memorize all the example. So it start with the root. So here in root, so in morphology, see, it's a very simple chapter, you just read, you understand. So I'm not going into very detail of it, but still I will highlight all the important points from where question is asked here. So in case of root, there are 3 types of root that you have to remember with example: taproot, adventitious root and fibrous root. What are the difference between them? What are the example? What are their characters? That is important. Then this regions of root, again in new NCERT it is, I think this is reduced, but still it is important. Here regions of root, there are 3 regions of root. First is the meristematic, elongation, maturation. You have to remember the characteristic of meristematic, elongation and maturation region. So here correct labeling is also important. Let's say you get the diagram and you have to label it. That also you need to understand. Then modification of root, in new NCERT it is removed. There is no modification of NCERT given. Then again in the stem is characterized by presence of node and internode. Node and internode is very important character. So here some examples are given, what is the characteristic of a stem, so that you can simply remember. Modification of stem is also removed here. Then coming to the leaf part. So leaf, the different part of the leaf, important here, that leaf has the axillary bud, it has petiole, it has lamina. These are the 3 main part. Then this petiole, lamina, leaf blade. So this labeling of the leaf here is important. Then inside the leaf there is venation, means arrangement of vein inside the leaf. It is of 2 types, reticulate venation and parallel venation. Reticulate in dicot, parallel in monocot. Further type of leaf. So here let me explain. Type of leaf, leaf can be simple or compound. If I say this is the leaf and this is its lamina. So lamina can be like this, or lamina also can be like this. Such kind of leaf is also found sometime. Now, if the lamina is not touching the midrib, this is the midrib. If lamina is not touching the midrib, it is simple leaf. But the moment the lamina touches the midrib, let's say it touched the midrib, then the moment it touched the midrib, now it becomes compound leaf, right? So the first basic difference between simple leaf and compound leaf is that the lamina of the leaf do not touch the midrib, it is simple. If it touch the midrib, it is compound. So this is point number 1. Point number 2, a compound leaf will have leaflets. A compound leaf will have leaflets. And here remember, it is also further divided into 2 types. One is called the pinnately compound leaf. Here there is a midrib, and through the midrib the leaflets are attached. When there is a midrib present and the leaflets are attached at different point on the midrib, it is called pinnately compound leaf. But when the leaflets are attached at the same point and the midrib is absent, then it is called palmately compound leaf. Remember, presence of rachis is a character of pinnately compound leaf. Absence of rachis is a character of palmately compound leaf. Example, very important, pinnately compound leaf is neem and palmately compound leaf is, here what is the example? Here it is bombyx. It is the palmately compound leaf. Here you can also see silk cotton is an example of palmately compound leaf. Is it clear? So pinnately and palmately compound leaf. Phyllotaxy, it is basically the arrangement of leaf. Arrangement, if it is alternate, China rose. It can be opposite, guava, or it can be whirl, as in Alstonia, right? Here examples are given, you just keep it in mind. Opposite, Calotropis, guava, whirled, Alstonia. Alternate, as in case of China rose, mustard and sunflower. Then further modification of leaf, again removed in new NCERT. Then inflorescence, very important. These are the 2 types of inflorescence. Inflorescence is basically the arrangement of flower on the floral axis. Arrangement of flower on the floral axis. Now, if the arrangement is such that the main axis continue to grow, then it is called racemose. If the main axis do not grow, then it is called cymose. So there are mainly three differences you have to remember. I will point out the three differences. First is the floral axis, whether it is limited or unlimited. Second is the flower position. Flower position. Third is whether it is acropetal or basipetal. So first, floral axis is limited or unlimited. What does that mean? Here, if you see the cymose, here the flower is present at the terminal position, means the axis is limited. So limited in case of cymose, unlimited in case of racemose. Now here the flower position, here flower position is terminal. In case of racemose, flower position is terminal. Here it is lateral. In case of racemose, it is acropetal. Acropetal means new flower at top. New flower at top. And basipetal means new flower at bottom. New flower at bottom. So this is the cymose and racemose. Simple difference, 3 points. Then coming to the main part, that is the flower. So flower, basically flower has 4 whorls. It can be unisexual, it can be bisexual. Flower can be complete, flower can be incomplete. If all 4 whorls are present, flower is complete. If any one whorl or more than one is absent, then it is called incomplete flower. Both androecium, gynoecium present, bisexual. Only androecium or gynoecium, so it is unisexual. So again, these parts are very simple. You can simply go through it. Now here, if this is one of the most favorite topic of NEET, every year one question is asked. Last year also one question was asked from here. This time also one question will be asked from here. So this is about the types of flower. So on the basis of position of whorls of flower, flower can be divided into 3 types: hypogynous, perigynous and epigynous. Now hypogynous means everything is below the gynoecium is hypogynous. So hypogynous, gynoecium occupy the highest position, other are below it. So such case the ovary is said to be superior, and the example is mustard, China rose and brinjal. Remember, mustard, China rose and brinjal is an example of hypogynous flower. Then perigynous flower. So this is also called as superior ovary. Now here, in case of perigynous flower, here you can see the thalamus itself has extended. So when the thalamus get extend but they do not meet at the top, it is called perigynous. So that's why it is called half inferior. Half inferior ovary. Right? So both B and C represents the half inferior ovary. But if this thalamus extend and get attached at the top, then it become epigynous, that is inferior. Inferior ovary. Now the example of perigynous is plum, peach and rose. Example of epigynous flower. Epigynous flower example is, here if you see epigynous, here sunflower, guava, cucumber, ray floret. So sunflower, guava, and ray floret. These are the simple example of epigynous flower. So 4 whorls of flower are calyx, corolla, androecium, gynoecium. Simple. So let's move ahead. So this is called the estivation. So estivation can be valvate estivation, twisted estivation, imbricate estivation and vexillary estivation. So here you have to simply remember all the example, right, which you all can easily read and remember it. Nothing is there to explain in it, but that is important. Question is asked from here whether through example or through the diagram. In the same way, here this is placentation. So these are the 5 type of placentation. You need to identify. Diagram will be given. For example, this is marginal, this is axile, this is parietal, free central or basal placentation. Question will be asked. Sunflower has. So you have to answer basal placentation. Pea has marginal placentation. Tomato has axile placentation. Mustard has, so parietal placentation. In that way, you have to remember the placentation, their diagram and example. Then in terms of, if we talk about androecium further, so androecium can form a single bundle, monoadelphous, 2 bundle, diadelphous, multiple bundle, polyadelphous. So here also example you have to remember: monoadelphous, diadelphous and polyadelphous. Example of polyadelphous: citrus, diadelphous pea, monoadelphous China rose. Fruit, here you have to remember the difference between these two and what is common. So they both are example of drupe fruit, but difference is that here mesocarp is edible, here seed is edible. Fruit of coconut is not edible. We do not eat the fruit of coconut. We eat the seed of coconut, and to be precise, we eat endosperm of coconut. We eat endosperm of coconut. Now here the fruit wall is divided into 3 parts. Epicarp, the outermost. For example, if we consider mango, so outermost thin peel is the epicarp. Middle fleshy part is the mesocarp. Inner tough part, hard part is the stony part is the endocarp. Then further the ovule develop into seed. In case of flower, ovule develop into seed and ovary develop into fruit, right? So here we will see the structure of seed. So this is a dicot seed, monocot seed. So dicot seed, 2 cotyledon are there, embryo is there, plumule is there, radicle is there. Then outermost seed coat is there, hilum, micropyle. This is the monocot. This is the monocot seed. Labeling of monocot seed is important here. You should be able to label it properly. Endosperm, eleuron layer, seed coat and fruit wall, scutellum, which is the cotyledon, coleoptile, plumule, radicle, coleorhiza. All are very important. So just see the labeling of this diagram. Now further coming to the family part. Now this is one of the very important part in which many of the students find it a little difficult. So see, regarding family, I will like to tell you one thing, that do not worry much about the families, right? Do not worry much means, of course, there is very less chance that many question will be asked from there. Even if it is asked, it is maximum 1 question, right? Then there is confusion that whether it is from new NCERT, old NCERT or something from outside. So see, there are 7 families in total. So all 7 families you have to go through, but just go through the floral formula and floral diagram. Don't get into detail about all, remembering all the characters and etc. Just the floral formula, what is symmetry, what is sexuality, how is calyx, how is corolla, how is androecium, how is gynoecium, and that is it. If it is epipetalous, then in addition to that, inflorescence, then fruit, monocot, dicot, or what kind of seed it is. That is it. Nothing more than that. You don't have to go and memorize 100 of character of each family, just that much. So here we will see the all those characters of all the family. So here the first family is the, if you see here, this is the Fabaceae family, right? So Fabaceae family here, if you see, so in case of this Fabaceae family, first of all, what is the symmetry? So symmetry here, as you can see, it is not a radial symmetry, it is actinomorphic symmetry. Then further, if you see, outermost wall is called calyx. So calyx has what aestivation? It has valvate aestivation. Corolla has a waxillary aestivation. Then you can see androecium. Androecium is 9 plus 1. 9 plus 1 is diadelphous. And inside is the gynoecium. So if I have to write the floral formula here, so it is actinomorphic, it is bisexual, then calyx is 5. Are they fused? Yes. Corolla, how many? Again 5, but they are given in 2 plus 2 plus 1. Then androecium, so it is 9 plus 1 diadelphous. Gynoecium superior 1, right? 1, and that is superior. So this is the floral formula, floral diagram, that is it. If this much you remember, it is fine. Now we will see some of the important part, like it is clearly bisexual, zygomorphic, 5 sepals. So just even if you remember this, all these points are covered here. So how many stamen is there? 10, they are diadelphous, right? Then ovary is superior. This is the floral formula. Clear? Then examples are important here. So example, all the pulses are example, edible oil is the example, dye is the example, fodder, ornaments, medicine, all are these examples. So these examples you simply remember. So I think this point I made very clear. Remember the floral formula, floral diagram and example. That is it. Nothing else is required. Further, if we see Solanaceae family. So Solanaceae family, again, if we observe this floral diagram, then we can simply say that this is a radial symmetry. Androecium, gynoecium, both is present. So it is bisexual in nature. Further, if we see how many calyx is present? 5. Are they fused? Yes. When the calyx is fused, it is called, gamosepalous. If it is not fused, it is called polysepalous. So here it is gamosepalous. Then further, if you see the petal, they are also fused. Androecium, they are 5. Now androecium is connected to the, androecium is attached to the corolla. So it is epipetalous condition. And then the gynoecium is also present. So you can see here epipetalous condition. When androecium is attached to calyx, this condition is called epipetalous condition, which is shown here. Then superior ovary, and there are 2 carpel or 2 pistil, right? Then again the example, tomato, brinjal, chili, belladonna, petunia, these all are examples. So just remember the example. So floral formula, floral diagram, example. Next, this family, Liliaceae family. So this is the first monocot family. Earlier two were dicot family. Here if we see, so here the floral diagram, if you see, it is a little unique. Although by seeing it, we can say it is actinomorphic symmetry. Both androecium, gynoecium is present, so it is bisexual. But here see, calyx and corolla you cannot differentiate. They both are fused together. When the calyx and corolla are fused, they are called perianth. So perianth is 3 plus 3, perianth. Then androecium, if you see here, 3 are arranged to outer radius, 3 are arranged to inner radius. So androecium we will say 3 plus 3. Now here all the androecium is connected to the perianth, so we will see this. It is called epitepalous condition or epiphyllous condition. Then this is superior ovary, and right, there are 3 pistils which are present and they are fused together. So 3 carpels are there, and this condition is called syncarpous. So we call it tricarpellary and syncarpous in nature, which is superior ovary. Here example again, remember, tulip, gloriosa, aloe vera, asparagus and colchicum. These all are the example of Liliaceae family. So these are all about the families which is mentioned in the now there are 3 more families. One is the Malvaceae family, Asteraceae family and Poaceae family. These 3 new families are also there that you also need to cover. Just go through the floral formula and the floral diagram with example. These 3 family also you need to cover. Now coming to the anatomy of flowering plant. So here again simple things are there. So just labeling the diagram, you just have to label it simply. Now here it start with the tissue. So plant tissue consists of 2 parts. It is meristematic tissue or permanent tissue. Meristematic tissue 2 parts, apical meristem, intercalary meristem, and the third one is the lateral meristem. Apical and intercalary is for primary and lateral meristem is for secondary growth, right? So then further, the permanent tissue, simple permanent tissue, 3 types, the parenchyma, collenchyma, sclerenchyma. Diagram you have to remember and their character you have to remember. Like parenchyma, isodiametric, store food, right? They are photosynthetic, they have intercellular space, all these character. In collenchyma, they do not have intercellular space, they have unevenly thick cell wall, right? They have pectin deposition at the corners. They are also photosynthetic. Both parenchyma and collenchyma are living, but the sclerenchyma is dead. The cell wall is thick, the cell wall is lignified, the protoplast is absent, right? So these are the difference. Then you have to read what is the difference between the sclerenchymatous fiber and the sclerid. Fiber and sclerid. So sclerid has a thicker wall, it has branched pit present, and it has elongated lumen present. Complex tissues. So it include the xylem and phloem. So xylem consists of trachea, vessel, xylem parenchyma and xylem fiber. Whereas the phloem will consist of sieve tube, companion cell, then phloem parenchyma and phloem fiber. Xylem, most of the xylem are dead except xylem parenchyma, and most of the phloem are living except the phloem fiber. Again, these are simple things, you can easily go through it. So as you can see, trachea, elongated, tapering ends are there, lignified wall is there. Now these tracheae are unicellular, whereas the vessels are here, vessels are multicellular. The unit of vessel is called vessel member. These vessel member are connected to each other with the help of perforation in their common wall. Then xylem fiber and xylem parenchyma. Xylem fiber will be sclerenchymatous. Xylem parenchyma, it will store food. Xylem parenchyma stored food in the form of starch, fat and tannin. Starch, fat and tannin. So again, these are simple points. Now here xylem can be divided into protoxylem, the smaller one, and metaxylem, larger one. So protoxylem develop into metaxylem. Now these xylem are arranged. So if protoxylem is toward inside, it is called endarch. But if protoxylem is toward outer side, it is called exarch. Endarch is present in stem, exarch is present in root. If we further talk about the phloem, so phloem consists of sieve tube element, it consists of companion cell, it consists of phloem parenchyma and phloem fiber. Now, the sieve tube element and companion cell are absent in gymnosperm. Instead, they have sieve tube cell or sieve cell and they have albuminous cell. Whereas here the companion cell have nucleus, sieve tube cell don't have nucleus. So companion cell control the sieve tube cell. The sieve tube cell and the companion cell, they both are attached with the pit field present in their longitudinal wall. Now here the companion cell also help in managing the pressure gradient inside the sieve tube cell. Further, remember this 2 point, phloem parenchyma is absent in most of the monocot, and phloem fiber, it is present only in the secondary phloem and it is absent in the primary phloem. So here also the the phloem also develop into smaller protophlloem and larger metaphlloem, but there is no exarch or endarch arrangement here. These tissue together form the tissue system. And tissue system is further divided into 3 types: epidermal tissue system, ground tissue system and the vascular tissue system. Epidermal tissue system consists of the monolayered epidermis like this, epidermal tissue system. Now these epidermal cell can be modified into stomata. They can have cuticle, they can have epidermal hair. This epidermal hair can be the stem hair, and it can also be root hair. Stem hair or root hair. So these are the modifications which are present in the epidermal tissue system. In the vascular tissue system, if we see, in the vascular tissue system, there is xylem and there is phloem. If they are present in 2 different radii, then they are called radial vascular bundle. If they are present on the same radii, then they are called conjoint. Conjoint can be of 2 types, closed or it can be open. When it is closed, then no secondary growth, and if it is open, then there is a secondary growth. Remember, radial vascular bundle is found in root, whereas in stem and leaf the conjoint vascular bundle is found. In case of dicot, the vascular bundle is open, and in case of monocot it is closed. In case of dicot leaf, the vascular bundle is also closed. Then finally, if we see the structure of root, so in case of root, if you see monocot, dicot root have somewhat similar structure, but there are 3 main differences. First is, here if you see in monocot, the xylem is polyarch. Here the xylem is not polyarch, only 4 or less than 6 is present here. Now here the pith is large. Here the pith is large in case of monocot, and monocot don't show secondary growth. Other than that, if we talk about the layer of the root, so outermost epidermis, right? Then the cortical layer, then the single layered endodermis, below it is the pericycle, then is the radial arrangement of vascular bundle, and at the center is the pith. In between the xylem and phloem, there is a special type of cell which is tissue, which is called conjunctive tissue. Right? Here the endodermis is surrounded by Casparian strip, which is made up of suberin. Similarly, further if we go with the stem, so in case of stem, you can see the outermost epidermis, hypodermis, hypodermis in case of, so this is dicot stem. So here the collenchyma represent the hypodermis. This parenchyma represent the cortical layer. And endodermis, right? So we can say that the cortex is divided into 3 sublayers. There are 3 sublayers which is present in the cortex. Inside the endodermis is the pericycle. Now pericycle here, if you see, pericycle here is semi-lunar in nature. Pericycle is semi-lunar and it is sclerenchymatous. Pericycle is sclerenchymatous. Inside the pericycle is phloem. Now since it is open, so in between phloem and xylem there will be a cambium. Then this phloem, this is a stem. So stem will have endarch arrangement. So you can see protoxylem is present inside, metaxylem outside, and at the center is the pith. So this is about the structure of the dicot stem. Now if we compare it with monocot stem, so here you can see the hypodermis. Now here the hypodermis is sclerenchymatous, but below that none of the layer you can see here. So can we say here the ground tissue is spread, or it is very large, and inside the ground tissue vascular bundle is scattered. It is scattered. Now in this case you can see vascular bundle are arranged as ring, but here they are scattered. Now also if you note, the larger one has the central position. The larger one has the central position and the smaller one has peripheral position. Further, the vascular bundle have small water pockets present in it, and these vascular bundle are surrounded by a bundle sheath over here. So these are the difference in character. So if I summarize, difference lies in the ground tissue, difference lies in vascular bundle, difference lies in the size of the vascular bundle, right? And of course this is stem, dicot, so it will show secondary growth. This will not show any secondary growth. This will have a open vascular bundle, this will have closed vascular bundle. Lastly, leaf. So leaf is simple, very simple. So here leaf, the difference lies in the upper adaxial surface, lower abaxial surface. So adaxial and abaxial surface in dicot leaf is also called dorsiventral leaf. The adaxial and abaxial surface are different because adaxial surface has less or no stomata, abaxial surface has more stomata. Toward the adaxial surface is present the palisade mesophyll. Toward the abaxial surface is present the spongy mesophyll. Spongy mesophylls have several air cavity present in it. Then the vascular bundle present in the dicot are of different diameter because they have reticulate venation. So veins are of different diameter, so vascular bundle also of different diameter. Whereas in monocot, all the veins are of the same because they have parallel venation. So except midrib, all the veins are similar. Further, here if we see adaxial, abaxial has equal distribution of stomata. Then here you see only one type of mesophyll cell is present. The diameter of vascular bundle is uniform, and also they have a special type of cell present toward the epidermis, which is called bulliform cell. Now this bulliform cell has this character that they manage the water condition of the plant. If present in plenty, they will be turgid and the leaf will be open. When the water is in scarcity, they will become flaccid, leaf will close. So this will help the plant to survive water scarcity. So this is about the leaf. So here, till here only syllabus is there. Secondary growth is removed from the syllabus. It is the deleted portion, so no need to cover the secondary growth. So anatomy, again it is a very simple chapter. We started with the meristematic tissue, then the permanent tissue, simple permanent, complex permanent tissue, then tissue system, finally the different organ of plant and comparison of monocot and dicot. So very theoretical part, just you have to revise, remember, that is it. Diagrams are important here. So that is about the anatomy. Now secondary growth, as I said, it is not a part, so this is old NCERT, so these are all given here. Now coming to the cell. So again this is important chapter, comparatively simple chapter in the sense that all of you must have been reading about cell for a very long time. So here also I will go through all the topic, but I will focus more on the important part. So here it starts with cell theory, which was given by Sladen and Schwann, right? Then it was further completed by another scientist who is Rudolf Virchow. Sladen and Schwann said that all living organism made up of cell or product of cell. Sladen and Schwann has said that all cell arises from pre-existing cell. Yes, hello Raj, how are you? So many of you are messaging also. So in this chapter, let me also know that in cell, all of you are okay with cell. Is it easy chapter? If it is easy, just mention easy. Or if you want me to explain certain specific part, then let me know. Just write in the comment. So here I was talking about the cell theory. So cell theory, very simple part, straightforward question is asked from here. Then next, overview of the cell. So this is also theoretical, in which different shape of the cell is mentioned. From here how question is asked. For example, it will be asked, what is the shape of RBC? What is the shape of WBC? Which cell has a branched structure, right? Which is the smallest cell? Which is the largest cell, longest cell, size of PPLO, size of mycoplasma, size of bacteria, size of virus? In this way they will ask straightforward question. What is the size of RBC, right? Vashvi is telling about ERN Golgi. Okay, fine. Yes, Vashvi, I will cover that in a little detail. Fine. So prokaryotic, now then after these, shape and size of the cell. So the shape and size of the cell mainly depend on what? It mainly depend on the function. Shape and size mainly depend on the function. So the prokaryotic, now we will move on to the prokaryotic and eukaryotic cell. So here if we see prokaryotic and eukaryotic cell, so first one, in prokaryotic cell there is no membrane bound cell organelle, means there will be no nuclear membrane, and all the membrane bound cell organ will be absent here. Instead, in the so what is present in the prokaryote? So first thing, the genomic DNA is present, and this genomic DNA will be circular. Double stranded, and they constitute only one chromosome. So if I ask you what is the number of chromosome, what is the number of chromosome present in bacteria? So the answer is 1. In all the bacteria only one chromosome is present. Whether it is E. coli, Bacillus or any other bacteria, it will always be 1. Then they have a extra genomic DNA, which is the circular DNA, which is called plasmid. Then they also have a mesosome. Mesosome is the membrane infolding. As you can see, this kind of infolding is present in the mesosome. It is membranous infolding and they have respiratory enzyme present in it. They have respiratory enzyme present in it. These respiratory enzymes helps in aerobic respiration. These mesosomes also play important role in cell wall formation, cell division, then distribution of DNA, DNA replication, and of course aerobic respiration. So these mesosomes are also called as the mitochondria of prokaryote because they help in the aerobic respiration. Apart from that, these can be present in the form of vesicle, tubule and lamellae. Then here if we further see, then there is another membranous infolding called chromatophore. So chromatophore contain pigment. Chromatophore contain pigment, and here chromatophore is found in the photosynthetic bacteria. It is found in photosynthetic bacteria because it has, see, prokaryotes also do not have chloroplast. So where the pigment is present? It is in chromatophore. So we can say chromatophore are the chloroplast of the prokaryote and mesosomes are the mitochondria of the prokaryotes. Then if we further see bacterial cell. So bacterial cell consists of filament, hook and basal body. Filament, hook and basal body. So this filament, hook and basal body, if this is the basal body, this is the hook and this is the filament. So filament is the longest part. Remember, in case of prokaryote, the flagella is made up of a protein called phloem protein. And then apart from flagella, they have pili and fimbriae. Pili is made up of pillin protein which help in conjugation and fimbriae help in attachment. So the surface have three outgrowth: flagella, pili and fimbriae. Then ribosome is present in prokaryote, and this ribosome is 80S, which has two subunit, 50S and 30S subunit. They have a role in protein synthesis, and in prokaryote they form polysomes. They form polysomes, also called as polyribosomes. Polyribosomes. Now, in case of eukaryotic cell, membrane bound cell organelles are present, so there is compartmentalization. Now, because there is compartmentalization in eukaryotic cell, so they will have a large number of membrane bound cell organelle. These membrane bound cell organelle have a special constituent which is same as the cell membrane. So what are the constituents? So they consist of lipid, protein, carbohydrate and cholesterol. So as you can see here, the lipid is present in the form of phospholipid bilayer. The protein can be integral protein or surface protein or peripheral protein. The cholesterol is present in between the phospholipid bilayer, and the carbohydrate is present on the surface of the membrane, is the carbohydrate. So these are the four components. Now, what is the most acceptable model to explain the membrane? It is the fluid mosaic model. It was given by Singer and Nicholson. He has used which cell to study it? RBC. And now what is the composition of RBC? 52% protein and 40% lipid is there. Now further, if we consider cell wall, so here cell wall, remember here 2 points. What is the difference in composition of cell wall of algae and cell wall of plant? So plant and algal cell wall has certain difference. So algal cell wall, cellulose, galactone, mannan and calcium carbonate is present, whereas in plant it is cellulose, hemicellulose, pectin and protein is present. Question is asked in the past regarding the difference in cell wall composition of algae and plant. Coming to endomembrane system. So as Vashvi has asked about endomembranes, about the ER and Golgi. So ER and Golgi both are a part of endomembrane system. So ER has a ribosome present. ER have two parts, RER and SER. See, RER is continuous with the outer nuclear membrane. RER is continuous with the outer nuclear membrane. On the surface of RER is ribosome present. Now this RER, since ribosome is present, it gives rough appearance and it helps in the protein synthesis. Whereas the smooth endoplasmic reticulum, ribosome is not present. Ribosome is not present. If ribosome is not present, then it will be smooth. If it is not present, then it will be smooth. The main function of RER is protein synthesis. Main function is protein synthesis and secretion. Here the main function is lipid synthesis. Here the main function is lipid synthesis. Apart from that, the steroid hormone synthesis is also the function. So remember here, structure and function. Two things you have to remember. What is the structure of RER, ACR, and what are their difference? That is it. So structure and function, both are clear for RER. Now RER, has it a role in secretion? Answer is yes. It helps in secretion, but intracellular secretion. Both intracellular and extracellular secretion is the function of Golgi. Now Golgi, if you see, it has two phases. It has cis phase and it has trans phase. From cis phase, vesicle enter inside. From trans phase, the vesicle goes out. Cis phase, trans phase. One is incoming, outgoing, convex, concave, right? So both the phases are interconnected to each other, but they are not similar. They are different but interconnected. Line of NCERT. It can be stained. It is densely stained, so it can be easily observed, discovered by Camillo Golgi. Now the protein and lipid come from the ER in the form of vesicle and joined to the cis part of the Golgi, and it comes out from the trans part. It comes out from the trans part. So this Golgi apparatus has three main functions. It has three main functions. First function is modification. Modification of what? Modification of protein and lipid which is coming from ER. These protein and lipid is converted into glycoprotein and glycolipid. And glycolipid. These glycoprotein and glycolipid are then the second function, packaging. They are packaged into a vesicle. And finally the third function is the secretion. So they are secreted, right? Now here the secretion is extracellular secretion. Extra and intracellular secretion, both is possible. Extra and intracellular secretion, both is possible here. Is it clear? So Golgi. Now this Golgi also has an important role in formation of cell plate, right? Golgi has this infolding. These are called cisternae. As you can see here, diameter of cisternae is also given 0.5 to 1 micrometer. That is the diameter. Then further, from the Golgi will arise the lysosome. So lysosome is also membrane bound and it has several hydrolytic enzymes present, lipase, protease and carbohydrase, which are all present here in the lysosome. These hydrolytic enzymes are active at the acidic pH. They are active at the acidic pH. Is it clear? So that is lysosome. Lysosome are also called the suicidal bag of the cell. They are called the suicidal bag of the cell. Vacuole is also vesicular structure. It is bound by the membrane. Inside the vacuole, the function is that they can store anything, means they have a general structure, they have a general function. The membrane of vacuole is called tonoplast. Membrane is called tonoplast. They have a role in their membrane have a role in active transport. So they have pump for different ions. Now these vacuole can be contractile vacuole, they can be food vacuole. Contractile vacuole help in osmoregulation and secretion. Food vacuole help in digestion of food and storage of food. These food vacuole and contractile vacuole both are present in amoeba and paramecium. So here vacuole, there is another term called gas vacuole which is found in prokaryote. That is not a true vacuole. Gas vacuole is also called as pseudo vacuole because it is not a true vacuole. Then coming to mitochondria. So mitochondria, so till here, remember all these are part of endomembrane system. What are the part of endomembrane system? The first part of endomembrane system is ER, Golgi, lysosome, vacuole. Then comes the next one, that is mitochondria. Now mitochondria, if we see the size of mitochondria, it is similar to the prokaryotic. This mitochondria has outer membrane, inner membrane, intermembranous space. The outer membrane is regular, but inner membrane has finger-like projection, and these projections are called cristae. Inside the inner membrane is present matrix, and in between the outer and inner is present the intermembrane space. Now here mitochondria play a very important role in ATP synthesis. That is why it is called powerhouse of the cell. Inside the mitochondria, ribosome is present. Ribosome is present. What kind of ribosome? 70S. It also have plasmid present. So it has its own DNA, it has its own ribosome, so it can synthesize its own protein also. So that is why it is called semi-autonomous cell organelle. Semi-autonomous cell organelle. Semi-autonomous cell organelle. Then, so here the main function is aerobic respiration. Now if I ask you, in which part of mitochondria Krebs cycle take place? So Krebs cycle take place in matrix. Where does ETS take place? So ETS take place in the inner mitochondrial membrane. Then plastid. Plastid is also a double membranous cell organelle. Here also we have outer membrane, inner membrane. Inside the inner membrane is the stroma. Inside the stroma is grana. Grana is the stack of thylakoid. These are connected together with the help of stroma lamellae. Now where is the pigment present? The pigment is present on the membrane of thylakoid. Right? So where does light reaction take place? Membrane of thylakoid. Where does dark reaction take place? In the stroma of thylakoid. Apart from chloroplast, what are the other types of plasmid? So other type of plastid. So plastid, it can be further two other type, chromoplast and leucoplast. Chromoplast has all pigment other than chlorophyll. Leucoplast has no pigment. So leucoplast has function of storage of food. Which food it can store? Chloroplast. It can store carbohydrate. It can store protein and it can store oil. When it store carbohydrate, it is called amyloplast. When it store oil, it is called elioplast, and when it store protein, it is called amyloplast. So these are the different types of plastids. Now inside the plastid also you will see ribosome, and these ribosomes are 70S. Inside they also have circular plasmid which is double stranded. So they are also called semi-autonomous cell organelle. Now ribosome, it was present in prokaryote also, it is present in eukaryote also. So in eukaryote both 70S and 80S ribosome is present. 80S is present in cytoplasm. It is present on ER, that is RER, and nuclear membrane. On all of this, in eukaryote it is 80S, but in where is this? In 70S in mitochondria and chloroplast. Chloroplast, or you can also say plastid. So it is 70S is present. So remember, where is 80S present? Where is 70? 70S has 50-30, 80S has 60-40 as the larger and smaller subunit. We have already seen that in prokaryote, ribosome can form polysome, means mRNA 1, several types of mRNA is one and several types of several ribosome attached, and they are doing the protein synthesis, right? Then cytoskeleton. So cytoskeleton, very simple part, you just have to remember there are three types of cytoskeleton present, that is microtubule, microfilament, intermediate filament. Question will be asked, which of the following is a cytoskeleton? So just these three terms you have to remember. Further, what is the function of cytoskeleton? So they have mechanical support, motility and maintenance of shape of the cell is the function of cytoskeleton. Is it clear? So cytoskeleton, nothing much is there. Now this is a little important because many of you get confused here. So let me explain this properly. See, we have seen that in flagella is also present in prokaryote. Now one thing which many students get confused is about 9 plus 0 and 9 plus 2. See, these both arrangement are not present in bacteria. In fact, bacteria have flagella, but they don't have microtubule. So they neither have 9 plus 0 arrangement nor they have 9 plus 2 arrangement. Clear? The structure of flagella is totally different in prokaryote and it is totally different in eukaryote. So here we are talking about the flagella in eukaryote. So flagella is present on the surface. So the part which is embedded in the surface is called the basal body, and the part which is outside, it is called exonym. So outer part is called exonym and basal body. So we are here seeing the structure of exonym, the outer part. So exonym, if we see, we have 9 peripheral doublets. There are 9 peripheral doublets present, and these doublets are interconnected with the help of a bridge called interdoublet bridge. So total how many? Answer it. Those who are listening carefully, answer me. How many microtubules are present in exonym? How many microtubules are present in exonym? Options are 18, 27, 9 or 16. A, B, C, D. Tell me, what is the exact number of microtubule present in exonym? Then what is the number of bridge? What is the number of bridge? So tell me the answer. So let's count here. If I say in exonym how many microtubule are present? So see, 9 doublet means 9 into 2. 9 doublet means 9 into 2, and at the center also there is 2. So 9 doublet 18 and 2. So it will be total 20. Although 20 I haven't given in option, so it will be 20, right? 20 is the option, is the correct answer. So 9 plus 2 arrangement, total number of microtubule present is 18 plus 2, that is 20. Is it clear? So there are 20 microtubules present. In the how many total bridge is present? So 9 at the periphery, 1 at the center. So total 10 bridges present. Question is asked. Then how many radial spoke is present? So radial spoke is 9 in number. Radial spoke is 9 in number. 18 is the peripheral microtubule. Peripheral microtubule. Peripheral microtubule. So understood this point? So 9 plus 2 arrangement is clear to all. 9 plus 2 arrangement is clear. It is of exonym, and 9 plus 2 arrangement show the 9 plus 2 arrangement show the arrangement of microtubule. Yes, Kavya Priya, right, your answer is correct, 20. Then further if we see basal body. So basal body of flagella is made up of centriole. Basal body is made up of centriole. Now this structure of centriole is not given in NCERT. So here just I will quickly draw it. So Now in this case they have triplet arrangement. They have such triplet arrangement. Now in this triplet, each triplet is connected to each other like this, right? So there is this triplet. Now at the center is the hub, and from the center is the spoke, right? So there is a central hub, then there is a radial spoke, then there is triplet, peripheral triplet. Overall this is called cartwheel structure. This is called a cartwheel structure. Now, so that's why it is called as 9+0 arrangement. 9 peripheral triplet and 0 central singlet. 0 central singlet. So this is the 9+0 arrangement. So 9+0 arrangement is of centriole. Centriole form the basal body of the flagella. Then coming to the nucleus. So nucleus is the one of the largest one of the largest organ of the cell. It has outer membrane, it has inner membrane, it has intermembranous space. So we can say it is also double membranous structure. In the nuclear membrane is present the nuclear pore. This nuclear pore connect the cytoplasm and the nucleoplasm. Here inside the nucleus is the nucleoplasm. Nucleoplasm have it is basically nucleoprotein, which is basically chromatin. Chromatin consists of protein, it consists of nucleic acid. Nucleic acid is RNA, DNA, protein is histone and non-histone. Histone and non-histone protein, right? Histone and non-histone protein. So overall what we can say that chromatin consists of histone, non-histone, RNA and DNA, all of this, because chromatin is nucleoprotein. Inside is also nucleolus. So what is the speciality of nucleolus? So nucleolus help in formation of rRNA. Nucleolus help in formation of rRNA. This rRNA, the rRNA of nucleolus is important. It is helpful in ribosome formation. That's why it is also called as the ribosome factory. Inside the chromatin can be of two types: euchromatin, heterochromatin. Euchromatin is functional, heterochromatin is non-functional or inactive, you can say. The euchromatin is lightly stained, loosely packed. Then heterochromatin is densely packed and it is densely stained also. When we talk about the chromosome, so the DNA pack themselves and form chromosome. Chromosome have centromere. It has centromere. Inside the centromere is the kinetochore. Each centromere has two kinetochore. The position of centromere will define the type of chromosome. If it is at the center, metacentric. A little away from center, submetacentric. Toward the end, acrocentric. At the end, it is telocentric. Now in that chromosome there can be satellite DNA also present, which represents the secondary constriction. Now this satellite DNA is non-staining, non-variable structure. It is constantly present. Now also it is not present in all the chromosome. It is not present in all the chromosome. So overall this is about the nucleus. Now last cell organelle is microbody. It is also membrane bound and it is present in both plants and animals. The two main example of microbodies are peroxisome and glyoxysome. Glyoxysomes. Clear? So this is about the cell. Now if we go to the cell cycle part, so cell cycle, the cell will grow and then the cell will divide. Now here again tell me, in cell cycle, rRNA full form. rRNA is ribosomal RNA. rRNA is ribosomal RNA. Actually the ribosome is made up of two parts, rRNA and protein. So you can say that the RNA present in the ribosome is called rRNA. There are several types of rRNA present in ribosome. So that is the rRNA. Now next, if we see cell cycle, so cell cycle basically is the sequence of event in which the cell grow and then ultimately it divide. Cell cycle consists of different stages: G1 phase, S phase, G2 phase, and then the karyokinesis, that is M phase. M phase is divided into karyokinesis and cytokinesis. Interphase is the long phase. It constitutes about 90 to 95% of the duration of cell cycle. M phase is divided into karyokinesis and cytokinesis. Karyokinesis is further divided into prophase, metaphase, anaphase and telophase. So here, which is the longest phase of G1 phase? Longest phase of interphase? It is the G1 phase. In G1 phase, RNA protein is synthesized. In S phase, DNA is synthesized and centrosome is synthesized. Centrosome and DNA is duplicated. In G2 phase, again RNA and protein is synthesized. So overall, if we say, then M phase is present between two interphase. Now during S phase, what happened? During S phase, the DNA content get doubled. Let's say it is 2C, then in S phase it will become 4C, and this will 4C, and then in the end it will again become 2C. Then in the M phase, prophase, what will happen? The nuclear membrane will disappear. As you can see, nuclear membrane disappear, chromosome condense, the chromatin condense and become chromosome. Then further, the centrosome, which was duplicated during S phase, start moving toward two opposite pole. Formation of spindle fiber will take place, and the cell organelle like ER, Golgi, they all will gradually disappear. So this is overall the event in prophase. Now in prophase, one important thing is there, and it is given as aster formation. In the prophase, it is given that there is aster formation. Somewhere it is given. So let's understand what is aster. So aster is the centrosome along with the radiating microtubule. The centrosome and radiating microtubule. Centrosome and radiating microtubule together constitute the aster. Now there are two poles, so two such aster are present. Two poles, so two aster are present here. Now along with aster, if we add the spindle fiber, then along with aster, if there is spindle fiber, then it is called mitotic apparatus. It is called mitotic apparatus. Is this clear? So two Aster along with the spindle fiber is called mitotic apparatus. What is aster? Centrosome along with the radiating microtubule is called the aster. Then coming to metaphase, the disappearance of the nuclear membrane mark the beginning of metaphase. The disappearance of nuclear membrane mark the beginning of metaphase. In this, what will happen? Gradually the spindle fiber will get attached to the centrosome. It will get attached to the centromere. Inside the centromere they will attach to the kinetochore and they will align the chromosome onto the metaphase plate. Now each chromosome at the metaphase plate is attached to both the poles. Each chromosome is attached to both the poles. Now during anaphase what will happen? Before anaphase, let's see metaphase. One more point, that in metaphase the chromosome are maximum condensed. So if you want to observe the morphology of the chromosome, you will have to go to metaphase. Then gradually the cell enter into anaphase. Here the centromere split. As the centromere split, the number of chromosome increases. So number of chromosome increase to double. So here let's say if I say in G1 phase the number of chromosome is 2n and DNA content is 2c. So in S phase it become 2n and 4c. In G2 phase it again become 2n 4c. Now in prophase it continue to be 2n and 4c. Right? In metaphase also it remain 2n and 4c. But when we get into the anaphase, here see, number of chromosome 1, 2, 3, 4. Here the number of chromosome is 8. So here the number of chromosome will be 4n and DNA content will be 4c, which will reduce to half here. So ultimately it will become 2n and 2c as it was initial. So what we conclude from here, that the number of chromosome during metaphase remain the same. Initial and final number of chromosome is same. So no changes in number of chromosome. So it is called, there is no number of, if there is no change in chromosome, so it is called equational division. It is called equational division, right? So there is one, is it possible to come 2c to 4c? See, this 2c will become 4c when there is a replication. S phase we have seen that there is a replication of DNA, and when there is a replication of DNA, 2c become 4c and it continue, and when again the cell divide, the 4c again become 2c. Then ultimately in the end cytokinesis will take place. Now cytokinesis in animal take place by furrow formation, but in case of plant there is a formation of a cell plate. Remember. So in case of plant there is cell plate formation, and in case of animal there is a furrow formation. Clear? Furrow formation and the cell plate formation. Further, so this is about meiosis. Now one important point you have to remember, meiosis mostly take place in diploid cell. It mostly take place in diploid cell, but there are some cases in which it also take place in haploid cell. For example, in animals, in the social insect. What is social insect? They are referring to bees. Right? So specifically male honey bee. Male honey bees. And in case of plants, it is lower plants. So in lower plants and male honey bee, mitosis take place in haploid cell, otherwise it mostly take place in the diploid cell. In mitosis, the number of chromosome remain the same, so it mainly help in growth and repair of multicellular organisms. So they have a role in growth and repair. Role in growth and repair. Now, what is the main difference between mitosis and meiosis. So in case of meiosis, what we observed? In mitosis, what we have seen, one replication, one division. So number of chromosome remain the same. In meiosis, what will happen? There will be one replication and two division. When one replication followed by two division, then it lead to one replication followed by two division, then the chromosome number will become half. This chromosome number become half in case of meiosis. So let's see, here two division is there, so both the division is divided into two part, meiosis 1 and meiosis 2. So let's see them one by one. So in case of meiosis 1, the first and foremost important stage is the prophase 1. Now prophase 1 is very long, it is divided into 5 stages. First one is leptotene. So here I will write leptotene. Then second one is zygotene. Then pachytene, diplotene, and finally dikinesis. Now let's understand here. Leptotene, you remember two main points, that chromosome become visible and chromosome compaction take place. Then zygotene. So in case of zygotene, what will happen? In case of zygotene, remember 4 points. Remember 4 points. The first point is there is formation of synapse. Right? There is synapsis. Second is formation of synaptonymal complex. Synaptonymal complex. Third is there is a formation of bivalent. And fourth is tetrad. So if question is asked, tetrad formation take place in zygotene, but its visibility will take place in the pachytene. So in pachytene, let's see what will happen. In case of pachytene, so there are 4 events here also. First, there is a formation of recombinants. There is a formation of recombination nodule. There is a formation of recombination nodule. Then there is also a formation of inside the recombination nodule there is enzyme recombinase. Then it will lead to the crossing over. And the last point is that the tetrad become visible. In case of pachytene only, tetrad become visible. Tetrad formation taking place in zygotene. Tetrad become visible in pachytene. Then comes diplotene. Here the synaptonymal complex disappear. It disappear. This synaptonymal complex disappear, followed by what? Separation of bivalent. And one important point is in diplotene there is formation of chiasmata. Chiasmata is what? It is the X-shaped structure. Chiasmata is the X-shaped structure. And finally the dikinesis. What happened in dikinesis? Terminalization. There is a terminalization of chiasmata. So overall these are the 5 steps in which you have to simply remember each of these points, right? Then it will be simple to memorize. Then after dikinesis, now here just one question, tell me if I ask you, in case of in this out of 5 stage, which can last for months to year? Which can last for months to year? So the answer is diplotene. In case of oocytes of several vertebrates, the diplotene can last for months to years. Then which is the last stage, or which is the transition to metaphase? So dikinesis is the stage which is the transition to metaphase. In metaphase, what will happen? So before this, let's understand, we are seeing the meiosis 1. So meiosis 1 G1 phase included 2n, 2c. Again here S will become 2n and 4c, because here replication take place. Then G2, 2n, 4c. Then in prophase, it remained 2n, 4c. Metaphase, now they are arranged as a bivalent, but there is no change in number of chromosome, so it remained 2n and 4c. Then anaphase, 2n, 4c. See here, in anaphase, no centromere split. If no centromere split, chromosome number remain the same. In mitosis, what we have seen, that the chromosome number become double, right? But in meiosis 1, chromosome number remain the same, and then at the telophase 1, when the dyad is formed, then the chromosome number become n and 2c. So can we say that the chromosome number reduced to half? So the chromosome number reduced to half. So meiosis 1 will be called as reductional division. Overall, meiosis is also called reductional division, and meiosis 1 is also called reductional division. Meiosis 1 lead to formation of two cell, which is called dyad of cell. What is the DNA content? 2n, and the dyad of cell will have not 2n, it is n and 2c is the dyad of cell. Then the cell will, this dyad of cell, so at the end of meiosis, there is a stage called dikinesis. This is interkinesis. It is interkinesis. In this interkinesis, what will happen? This interkinesis is different from interphase. This is a short phase and no DNA synthesis. No DNA synthesis. Further, interkinesis, then the cell enter into meiosis 2. Now meiosis 2 is exactly same as mitosis. Meiosis 2 is exactly same as mitosis. So here if we see, see, the cells in dyad were N and 2C. So N and 2C cell enter into meiosis. So in prophase, same thing happen, nuclear membrane disappear, centrosome move toward the opposite pole, the cell organelle gradually disappear, etc. Now in prophase, it will remain N and 2C. Then in metaphase, N, 2C. Now tell me in anaphase what will happen? In anaphase, there is a splitting of centromere. So if there is a splitting of centromere, will the chromosome number become double or not? So the answer is yes. Here the chromosome number become double, so N will become 2N and 2C, right? And then finally after telophase, what is formed? Tetrad of cell. Tetrad of cell. So this tetrad of cell has DNA content N and C. So here you see in meiosis 2, it started with N and it end with N. So is there any change in number of chromosome? So can we call meiosis 2 as equational division? So remember this point also, that meiosis 1 is called reductional division, meiosis 2 is called equational division, because in meiosis 2 there is no change in number of chromosome. Meiosis 1, the chromosome number become half. On the other hand, in meiosis 1, the DNA content remain the same, 2C remain 2C, but here 2C become half. So can we say in meiosis 2, the chromosome number remain the same, but DNA content become half. So this is the difference. Overall, meiosis is reductional division. Meiosis 1 is reductional division, meiosis 2 is equational division. Mitosis is also equational division. Lastly, the significance of meiosis. So we can clearly see that meiosis is reducing the chromosome number by half, right? Reducing the chromosome number by half. So this is very important to maintain the same number of chromosome in next generation. If chromosome number don't reduce by half, then next generation will have double the number of chromosome. See, 43 become 92, right? So it is reducing half. 46 become 23, 46 become 23, then 23 plus 23, 46. So next generation also 46. So meiosis is basically helping in conservation of number of chromosome. So here a word is given, paradoxically. Paradoxically means that it is contradictory, right? When something contradicts, it is called paradox. Means it appears as if it is reducing the number of chromosome by half, but actually what it is doing, it is maintaining the same number of chromosome by reducing the chromosome number by half. So that is why it is called paradox. Now in this meiosis, in case of meiosis, there is a crossing over. So there is a genetic variability which will lead to the evolution. Genetic variability will lead to the evolution. So hence, now this part is covered. So now if you have to summarize the difference between mitosis, meiosis, you can easily summarize. Now one or two question I would like to ask here. Let's say I'm asking this question. So now the first question here I am asking, all of you who are attending here, just see, if I am telling you that a leaf cell has number of chromosome, a leaf cell has number of chromosome 16, then number of chromosome during S phase, during anaphase, during telophase, during G2 phase of mitosis. So A, B, C, D. Tell me the number. What will be the number of chromosome during A, B, C and D? If the number of chromosome in leaf cell is 16 and during mitosis, the cell is undergoing mitosis. So tell me the number of chromosome during S phase, anaphase, telophase and G2 phase. So all of you tell me the answer quickly. So you have 2 minutes. Tell me the answer. So I guess all of you must have done it. So I can see all has 16 chromosome. Okay. 2n is in anaphase. Okay. Tell me the exact number. Exact number. So Sudhakar has given me this 16 chromosome. All has 16 chromosome. Okay. So let's see the answer. Let's see the answer. So leaf cell has number of chromosomes 16. So number of chromosomes 16 means 2n is given as 16. Now during the mitosis, I am writing here in different G1 phase the number of chromosome 2n, DNA content 2c. In S phase 2n 4c, G2 2n 4c, prophase 2n 4c, metaphase 2n 4c, anaphase. What happened during anaphase? The centromere split. When the centromere split, then 2n become 4n 4c, and finally telophase, cell divide, right? Telophase and cytokinesis, cell will divide, so it will become 2n and 2c. It will become 2n and 2c. So it start with 2n and 2c, it end with 2n and 2c, right? So now let's see on this basis if I have to answer it. Tell me, S phase number is 2n, so 2n is, so it will be 16. Anaphase, anaphase 4n, anaphase is 4n, so when it is 4n means it will be 32. Telophase, again 2n, so 16, and G2 2n, so it will be again 16, right? So so Sudhakar, so your answer was to some extent correct, but only this part, anaphase, right? Vashvi also said all 16. So Vashvi, I guess now you understood that why it is 4n here, because during anaphase, always remember the number of chromosome become double. So if the number of chromosome become double, so it is 32. So let's move ahead. So I think this part is clear. Now in the same way, if question is asked about the DNA content, again you will use the same rule. This table is important, you just keep it in mind. You can note it down, you keep it in mind. In the same way, for meiosis also there can be a table, so that also let me write it this side. It will help you in solving all kind of question. So in meiosis, if I say, let's say G1, it is 2n and 2c, right? S 2n 4c, G2 2n 4c. Then here prophase 1 2n 4c, metaphase 1 2n 4c, anaphase 1 2n 4c, right? See here, in anaphase 1 no change in number of chromosome, note it down, and then finally telophase 1 it become n and 2c. So this is about the meiosis 1. This is meiosis 1. And then in meiosis 2 what will happen? Prophase 2. So here n 2c. Metaphase 2 n 2c. Anaphase 2. Now anaphase, so here again centromere split, so it become 2n 2c. And finally in telophase 2 it become n and c. So this is for meiosis 2 and this is for meiosis 1. So this table also you can keep it in mind, and this table also you keep it in mind. Both will be very helpful to you to answer all the number based question from this topic. So now let's move ahead. So now next chapter is biomolecule. So biomolecule again is a bit confusing chapter to many, because there are few things which are which student find it a little difficult to memorize. So here I will highlight you all the points which you will have to remember, and few points are there to understand. So let's understand here. First, you have to remember these tables, because from these tables sometime questions is asked that which is the most abundant element in earth crust, in human, right? What are the different inorganic constituent present? See, water is also an inorganic constituent which is present maximum in our body. Many times students do the mistake of considering water as organic. Water is inorganic constituent of the body. So here, let me summarize this entire part. So here the first concept is about the how you isolate and what is present in acid soluble and what is present in acid insoluble. So when you take the any tissue, you crush it, you take out the biomolecule, then you will get two part. You will get two part. One is there as the filtrate. Here is the filtrate. This part is the filtrate, and this part is called retentate. This retentate is also called acid insoluble fraction. Insoluble fraction, and this one is acid soluble fraction. See, now, so till this part things are clear. Then beyond this, if I say, then in the acid insoluble fraction, in the acid, this is the soluble fraction, this is insoluble fraction. So here all the macromolecule is present, and here micromolecule is present. Micromolecule is present. Exception is there. Exception is what? Exception is lipid. Lipid, although is a micromolecule, present in the macromolecule. Clear? It is present in the, along with the macromolecule, in the acid insoluble fraction, because lipid is also insoluble, so it will remain with insoluble. So that is one exception. Then these macromolecule include both organic constituent and inorganic. They constitute both organic and inorganic. Now here in case of retentate, if I further see, so they mainly consist of protein, carbohydrate, right? Then they consist of polynucleotide. So these are the main macromolecule, and of course lipid is also a constituent, but protein, carbohydrate and polynucleotide, they are all present in the retentate. And whereas if I say amino acid, right, glucose, all are present in the filtrate. Here it is given they have a mass of 10 of the range of 10,000 Dalton. On the other hand, the mass of lipid is always less than 800 Dalton. So you can clearly see that it is a micromolecule but present along with macromolecule. Now how will you distinguish between macromolecule and micromolecule? So see, the threshold is 1000 Dalton, right? Anything less than 1000 Dalton is called micromolecule, more than 1000 Dalton is called macromolecule, right? So three numbers are there: protein, carbohydrate and polynucleotide are of the range of 1000 Dalton, of the range of 10,000 Dalton. But in general, if we say macromolecule in general, they should be more than 1000 Dalton. If less than 1000 Dalton, then micromolecule. And for lipid we say not more than 800. So it is anyway less than 1000, so it is micromolecule. Is this point clear totally? So there is a retentate, there is filtrate, then in the retentate you have all the macromolecule, in the filtrate you have micromolecule. Clear? So yes, Sudhakar, I will cover all the biomolecule. I will not miss any of it which is there and which will be asked in the test. Don't worry. So here this part I guess you understood that how the biomolecules are divided in between retentate and filtrate. This point is clear. Now then it starts with amino acid. It starts with amino acid. Now in amino acid it is a derivative of methane. Methane is CH4. So it is also derived from CH4. Here actually what happened, on one side it is COOH, other side it is NH2, then it is R group, H group. So it has four arms. One is called the carboxyl group, amino group, R group which is the alkyl group or variable group. So these are the four branch of amino acid. Now characteristic, these three are same in all the amino acid. So what will change? Only changes in the alkyl group, right? So alkyl group is the variable group. The characteristic of amino acid depend on the alkyl group. How many total amino acid are there? There are total 20 amino acid present in protein. See, actually there are hundreds of amino acid, but present in protein is only 20. So there are 20 amino acid present in protein, right? And these 20 amino acid will differ in their alkyl group. Then here it is mentioned that these alkyl group, when it is hydrogen, then it is glycine. When it is methyl group, it is alanine. When it is methyl group, it is alanine, and when it is hydroxymethyl, it is serine, as you can see here. See, when it is hydrogen, it is glycine. When it is methyl group, it is alanine, and when it is CH2OH, that is hydroxymethyl, then it is serine. Question will be directly asked here with diagram also. Then further, if we see this, these are the branch of amino acid. Now this part can lose H plus and become COO minus, and this part can gain H plus and become NH3 plus. Now what happen if both things happen side by side? Means this attain a negative charge and this attain a positive charge. In that case, that will be called as the Zwitter ion. So how will you identify a Zwitter ion? On one side it has a negative charge, other side it has a positive charge, so it is called Zwitter ion. Clear? Now further, this amino acid can be further divided into acidic amino acid, basic amino acid, aromatic amino acid, etc. So there are some of the examples given which you need to remember. See here, acidic amino acid is glutamic acid. Whenever acid is in the end of any amino acid, of course it is acidic amino acid. Basic amino acid, it is lysine. Remember, lysine is basic amino acid. Even arginine is also basic amino acid. Then neutral amino acid. Neutral amino acid is valine, right? Neutral amino acid is valine, whereas here you see aromatic amino acid. Aromatic amino acid is tyrosine, tryptophan and phenylalanine. Phenylalanine. So here this will be asked in question this way: which of the following is aromatic, which of the following is is the neutral, basic, acidic, right, which has a side chain of hydroxymethyl. So all you can simply answer. So this is about basics about the amino acid. Is it clear? So amino acid is covered. Then next is the fatty acid. So fatty acid basically has so if we talk about fatty acid, so fatty acid simply is this, you can see. Before that, let's understand when we talk about fat. So fat consists of simple fat and it consists of complex fat. Simple fat include fatty acid, it include glycerol, and further it include triglyceride. They all will come under simple fat. In complex will come the phospholipid, lecithin, cholesterol, right? So in the end, first is given fatty acid, glycerol, then triglyceride. All three are part of simple fatty acid, and then these are the part of complex fatty acid. So simple fatty acid is simply RCOOH, where R is again the alkyl group. As you can see in palmitic acid, this R has how many carbon? This R has 15 carbon. So 15 plus 1 is 16 carbon. So if question is asked, how many carbon palmitic acid have? So 16 including carboxyl carbon, but in alkyl group there are 15 carbon. So now this is fatty acid. Then glycerol. Glycerol is this having 3 hydroxyl group. You can see CH2OH, CHOH and CH2OH. So this is glycerol. Then this fatty acid and glycerol will come together and form triglyceride. If one fatty acid, one glycerol, monoglyceride. Two fatty acid, one glycerol, diglyceride. One glycerol and 3 fatty acid, triglyceride. So always remember, mono, di, triglyceride depend on the number of fatty acid. In each case, one glycerol is only required, but number of fatty acid will determine whether it is mono, di or triglyceride. So in the formation of triglyceride, which bond is formed? So here ester bond is formed. In this case, ester bond is formed. So monoglyceride, how many ester bond? 1. Diglyceride 2, triglyceride 3. So number of ester bond will depend on the number of fatty acid. So as you can see here, 3 ester bond is present. This is triglyceride. Now in some cases, so this is all the 3 simple fat. How complex fat is formed? When if the fatty acid is replaced by one of the phosphate group, then it is called the phospholipid and it forms the complex fat, right? So phospholipid, one of the fatty acid is replaced by a phosphate group. Further, this is so this is the structure of lecithin, which is a type of phospholipid. Here we see this is cholesterol, right? Cholesterol having 4 ring and these are the 6 carbon here. So you can identify cholesterol, you can identify lecithin, triglyceride, glycerol, fatty acid. All you can easily identify. Then, so this is about the fat and fatty acid as it is mentioned. Now here one more point is mentioned is about what is the difference between fat and oil. So fat is usually solid, oil is liquid. Fat is having so oil is having lower melting point, fat is having higher melting point, right? So here if we further say, so fat remain solid at room temperature, oil remain liquid at room temperature, right? So now, so fat is obtained from which source? Animal or plant? So it is usually from animal source and oil is usually from plant source. So these are the difference between fat. Now the alkyl group of fatty acid, they can be saturated, unsaturated, monounsaturated, polyunsaturated, depending on number of bond. If double bond is present, unsaturated. Then here we are talking about the proteins, right? So before that, okay, first let's cover the protein part, then we will go to the primary and secondary metabolites. So first cover the protein part. Here amino acid, we have already seen amino acid. So amino acid will come together to form peptide bond. This peptide bond is formed between the carboxyl group and amino group, forming a peptide bond. These peptide bond together form a polypeptide, right? This is polypeptide which is formed. These polypeptides have several peptide bond, and the polypeptide have several structure. For example, one, it is primary structure. Protein can have primary structure. Then they can have secondary structure, which can be helical or sheet-like. Helical, hydrogen bond is present, right? So in primary, there is no hydrogen bond present. In secondary structure, there is hydrogen bond present. In tertiary structure, along with the hydrogen bond, there are many other bonds present, which is called tertiary structure. What are the other bond present? Disulfide bond. Tertiary structure. So here disulfide bond present. Disulfide bond is present. Clear? Then several tertiary structure will come together to form a quaternary structure. So these are the point as shown here also. See, primary structure, secondary structure, tertiary structure and quaternary structure. You can see here in tertiary structure disulfide bond. In the secondary structure there is hydrogen bond, and in quaternary more than one tertiary structure is coming together. So there are four level of structure in case of protein: primary, secondary, tertiary, quaternary. Which is the functional form? So tertiary and quaternary are the functional form. Which are non-functional? The first two are non-functional, right? Non-functional form. Quaternary example given is hemoglobin. Hemoglobin has 2 alpha. Hemoglobin has 2 alpha and 2 betas structure. So total 4 subunits are present here. Now further, here as protein is there, similarly polysaccharide more than so 2 monosaccharide will come together, and which bond here will be formed? Here glycosidic bond will be formed. Glycosidic bond is formed. In protein peptide bond is formed. Here glycosidic bond is formed. Now in this glycosidic bond, in here we have to, in polysaccharide we have to study, see, starch, glycogen, cellulose and inulin, right? These are the main polysaccharide which is given. So starch will have glycosidic bond. Starch is branched, right? Starch is, it has linear as well as branched structure also, right? So then glycogen is also branched. Cellulose is straight. There is no branching in cellulose. So what happened? Because of its helical structure, iodine get trapped in the helices, and hence starch give positive iodine test. Starch give positive iodine test. Glycogen, it is branched, but it is the storage form found in animals, right? Starch is the storage form of sugar in animal. The glycogen is the storage form of sugar in animal. Starch is the storage form of sugar in plants. Cellulose is not digested by animal. Cellulose is only digested by the ruminant animal, that is the cattle, they can be digested. Inulin is the polymer of, it is the polymer of fructose. So remember, all of them are homopolysaccharide. All of them are homopolysaccharide. The first three, cellulose, starch and glycogen, are polysaccharide of glucose, whereas inulin is the polysaccharide of fructose. It is the polysaccharide of fructose. Now, if you see here, the linear chain has 1,4 glycosidic bond. 1,4 glycosidic bond means between first carbon and fourth carbon, but branch has 1,6 glycosidic bond. Branch has 1,6 glycosidic bond. Overall, in the end, there is one special type of compound called amino sugar is there. Amino sugar. So remember, it is also a monomer type, and it give rise to complex polysaccharide. Here chitin is also mentioned, which is also a homopolymer, right? Chitin is a homopolymer of amino sugar. Nucleic acid, this we will see in the molecular basis of inheritance in detail. So here we are not talking about it. Then let's talk about the enzyme. So enzyme, basically what they do, enzyme increase the rate of reaction by decreasing the, it increase the rate of reaction by decreasing the activation energy. So substrate get converted into product, and in that process they reduce the activation energy. They reduce the activation energy. So this activation energy is reduced, and due to that the substrate is converted to product. So question, how does an enzyme increase the rate of reaction? By decreasing the activation energy, right? Then enzyme bind to the substrate and form enzyme substrate complex. These enzymes do some change in bond of the substrate and get converted into enzyme product complex, and then the product is also released, enzyme is also released. This enzyme will again be used in the formation of product. So enzymes are not consumed in the reaction, they take part in the reaction. So this is how. So energetically this is very important from exam point of view, and this 4 points are also important. From here question is asked that what is the correct sequence of event in the action of enzyme, that also you need to cover. Then there are certain factors, enzymes, there are certain factors which are responsible for enzyme activity, and those factors are temperature, pH and substrate concentration. Now here the substrate temperature, it will form an inverted bell, so it forms a bell-shaped curve in which at an optimal temperature the enzyme activity is maximum. Beyond that or after that the enzyme activity will reduce. Same pattern is followed by the pH. In substrate, what happened? As the substrate concentration increase, the rate of reaction will increase, and then it will gradually become constant. So this is the saturation. Now this is called Vmax. Now half of the Vmax by 2. So that Vmax by 2 is called Km. So Km is the substrate concentration at which the rate of reaction is half of maximum, right? So as we increase the substrate concentration, initially the rate of reaction will increase, then gradually it will become saturated, which is called Vmax, that is maximum velocity. Km is the concentration at which the rate of reaction is half of maximum. Why it happens? Because here number of enzyme is more than substrate, here number of enzyme is equal to substrate, and here the number of substrate is more than enzyme, and that is why this difference is there. So this is how it is affected. Then enzyme which has the active site, let's say this is the active site of enzyme and there is a substrate. So substrate will go and bind to the enzyme, but many time what happen, let's say some inhibitors are there. which also compete with substrate to bind, and when inhibitor bind to the active site, then the substrate will not bind and this will lead to inhibition. So here remember 3 points are given. One is enzyme. Which enzyme? Example of competitive inhibition. Which enzyme? Succinic dehydrogenase. What is the substrate? So the substrate is succinate. And what is the inhibitor? Inhibitor is malonate. See here, what is very important is remember which is enzyme, which is substrate and which is inhibitor, because students usually do confusion in this. Now here you can easily go through it, what are the enzyme, and you can easily identify types of enzyme based on their bonds. Further, most of the enzyme are made up of protein, but there are enzyme which has some non-protein part also, and these non-protein part of the enzyme is called cofactor. This cofactor can be of 3 type. Cofactor can be of 3 type. First one is called prosthetic group. Second one is called coenzyme. And metal ion. Prosthetic group, what is it? So it is organic. Prosthetic group is organic and it is tightly bound. Example is heme group, right? Coenzyme, so it is organic, it is loosely bound, and for example, vitamin, niacin, NAD and NADP. These are example of coenzyme. Then metal ion. Metal ion are inorganic, they are tightly bound, and the example given here is zinc, which is present in carboxypeptidase. If cofactor is not present, then enzyme will become totally inactive. Question was asked in 2024 regarding the carboxypeptidase. Then here in plant physiology, so here what is the main topic where you find it difficult? So here light reaction is very important. In this, first of all, the pigment was studied, and here this is the absorption spectra, action spectra and overlapping spectra. So in the absorption spectra, chlorophyll A, B and carotenoid are having the absorption spectra. Here we find out that what are the peak of absorption shown by different pigment. Then here is at which wavelength photosynthesis is maximum, and this overlapping of action and absorption spectra give us the idea that chlorophyll A is the major photosynthetic pigment. So what is the purpose of overlapping? Find out which is the major photosynthetic pigment. Now here also many of you find it a bit confusing that there is a reaction center, right? Surrounding the reaction center are the accessory pigment. These accessory pigments are called antenna. These are called antenna, also called as light harvesting system, right? You call it antenna or call it light harvesting system or call it accessory pigment. See, one confusion is there. Don't consider that a light harvesting system is equivalent to light harvesting complex. No, light harvesting system is light harvesting complex minus reaction center. If you include reaction center in light harvesting system, it will become light harvesting complex, right? So these all are representing the accessory pigment. Now when you add reaction center to it, then it become light harvesting complex, also called photosystem. And this photosystem is of two type, PS1 and PS2. PS1 absorb at 700 and PS2 absorb at 680 nanometer. So this is about the light harvesting complex. Then these light harvesting complex trap the energy, trap the sunlight, and they take part in electron transport. So as we can see here, they will take part in photophosphorylation, which is of two types, non-cyclic and cyclic photophosphorylation. Now if you see the non-cyclic here, LHC, which is the LHC here, photosystem 2, uphill electron acceptor, downhill PS1, again uphill, and finally electron is accepted by NADP plus. So oxygen is released, ATP is formed, and NADPH is formed. So these are the three products: oxygen, ATP and NADPH. Out of this, ATP and NADPH is used in the dark reaction. What is the source of electron? So H2O is the source of electron. Splitting of water will provide electron, which is taken by NADP plus and NADH is formed. Further, there is also a non-cyclic there is a cyclic photophosphorylation. Here electron excited from photosystem 1 come back again to photosystem 1. When they come back to photosystem 1, then there is no source of electron required. So no splitting of water, no oxygen, no NADPH formation, only ATP formation, right? So here remember the difference between non-cyclic and cyclic photophosphorylation. This electron transport system will lead to the chemiosmotic hypothesis. This hypothesis explains the formation of ATP in both cyclic as well as non-cyclic. Although here it is shown cyclic, but similar pathway is there in the non-cyclic also. Here what happened? As you can see, electron is taken by PS2. Electron is excited to plastoquinone. From there it goes to B6F to PC. Then it goes to PS1. Then it goes to ferredoxin, FD, FNR, and finally accepted by NAD plus, and it becomes NADH. So this is the pathway of electron. As the electron move, H plus is passed inside the lumen. Here so H plus is accumulating inside the lumen. So high concentration inside, low concentration outside. So electrochemical gradient is created. Due to this electro gradient created, H plus will cross the ATP synthase. As it cross, ATP is formed. So this chemosynthetic pathway actually give us an idea about the ATP formation, how ATP is formed. So what are the requirements? Membrane is required, a proton pump is required, ATP synthase is required, right? And then remember the position where splitting take place, where NADPH is formed, which enzyme is involved, right? Which side ATP is formed, which side the movement of H plus is there. So all those points question is asked. Then once the NADPH and ATP is formed, these are used in the dark reaction. Where does dark reaction take place? In the dark reaction take place in the stroma. The light reaction we have seen, it take place in the membrane of thylakoid. Dark reaction take place in the stroma. Here the ATP and NADPH synthesized in the light reaction will be used. So what will happen here? CO2 is fixed using the product of light reaction. So here who will first react with CO2? So RUBP. RUBP with the help of enzyme Rubisco. Rubisco will form 2 molecule of PGA. This 2 molecule of PGA will form 2 molecule of triose phosphate. So this step occur twice, and then regeneration into RUBP. So this 6 carbon again become 5 carbon. This is 5 carbon. 5 plus 1, 6 carbon, right? Means 2 PGA, so 3 carbon, so 2 into 3 carbon. So 6, here also 2 into 3 carbon, 6. Now 6 become 5. So where does 1 carbon go? So 1 carbon is fixed. 1 carbon is fixed here. So in each cycle, how many carbon is fixed? 1 carbon. In each cycle, how many ATP and NADPH is required? So in one cycle, 3 ATP and 2 NADPH is required. Now, and 1 CO2 is fixed. 1 CO2 is fixed. Now, if question is asked that to fix the glucose, how many to fix one molecule of glucose, how many ATP will be required? So it will be 18 ATP and 12 NADPH, right? So one cycle 3 ATP, 2 NADH, 1 glucose, 18 ATP and 12 NADPH. Now here the problem is Rubisco enzyme has affinity for oxygen also. So CO2, Rubisco will react with CO2 also and oxygen also. So photorespiration will take place. To avoid that, C4 plant develop. Now here no photorespiration because they have Cran's anatomy. Here CO2 will react with phosphoenol pyruvate and it forms C4 acid, that is oxaloacetic acid. These oxaloacetic acid will get converted into malate or aspartate, and this malate aspartate enter into the bundle sheath cell. Here it will release CO2 and become pyruvate or pyruvic acid. This pyruvic acid transported back regenerate into phosphoenol pyruvate. So here it is very much clear that since oxygen and Rubisco are separated, so there is no photorespiration. This is more efficient, but it is more energy consuming. So here one glucose molecule formation will require how many ATP? 30 ATP. And how many NADPH? 12 NADPH, right? So in C3 18 ATP required, here 30 ATP is required. So isn't it more expensive? But it is also more efficient. So photorespiration, it takes place only in the C3 plant. Means out of C3 and C4 it takes place in C3 plant, C4 plant no photorespiration take place. What happened here? So here RUBP react with oxygen and form 2 PGA. Usually 2 molecule of PGA is formed. Here only 1 molecule of PGA is formed and 1 molecule of 2-phosphoglycolate is formed, right, which is 2 carbon. That's why it is also sometimes called C2 cycle. In this process, so RUBP, this is showing what happened during photosynthesis, but during photorespiration, this instead of 1 molecule of PGA, 1 phosphoglycolate is formed. See here, 2 carbon phosphoglycolate is formed. In this process, no ATP released, no NADPH released. What is released? CO2 is released and O2 is consumed. So it is just like respiration. It takes place in 3 cell organelle: chloroplast, mitochondria and peroxisome. It takes place in high light condition, right? It takes place in high light condition, and it also takes place in low CO2 concentration. If CO2 concentration is high, then no photorespiration will take place. Now here if we see what are the different factors which affect the photosynthesis. So here light. So light is rarely a limiting factor. What is limiting factor? When any function depend on more than one factor, then factor which is present in least amount is called the limiting factor, right? So here light is rarely a limiting factor. And what is that? So why is so? Because plant will become the photosynthesis will become saturated only at 10% of the light intensity. So it is rarely a limiting factor. 2 examples are there. It is limiting for plants found in shaded area and plant found in dense forest, right? So here if I say what is the light intensity, what does D represent? It represents 10% of light intensity. What does E represent? It represents the maximum rate of photosynthesis, right? C represent constant rate of photosynthesis, A represent increasing rate of photosynthesis. So second is carbon dioxide. So carbon dioxide carbon dioxide is the most common limiting factor. It is the most common limiting factor. So in this most common limiting factor, C3 plant has the saturation concentration of 450 microliter per liter, and C4 plant has saturation concentration of 360 microliter per liter. Atmospheric concentration is 400 microliter per liter. So due to this, currently the concentration of CO2 is limiting for C3 plant and it is non-limiting for C4 plant. It is non-limiting for C4 plant. So if I increase the CO2 concentration, who will be benefited? C3 plant. Who will not be benefited? C4 plant. Non-limiting for C4 plant, limiting for C3 plant. Then what is the toxic concentration of CO2? So it is 0.05%. That is the toxic concentration. Talking about temperature, so C3 20 to 25 degree, C4 30 to 40 degree, right? Then water. So in case of water do not directly affect the rate of photosynthesis. It indirectly affects the photosynthesis, because what will happen in case of water, if there is scarcity of water, then there will be closing of stomata, there will be wilting of leaf, and due to that availability of CO2 will be less. So water affect but not directly, it indirectly affect the rate of photosynthesis. So this is all about the photosynthesis. Now talking about respiration. So in respiration, it has two branch, aerobic and anaerobic respiration. So in both the case, which is the most common step? So the common step is glycolysis. It takes place in both aerobic as well as anaerobic respiration. So in the glycolysis step, if we see here, it is a 10-step process. In glycolysis, overall, how many ATP is produced? So if you see here, 4 ATP is produced, but how many get consumed here? 2 ATP is consumed. So net ATP produced is 2 ATP, and these 2 ATP is the substrate level ATP. Apart from that, 2 NADH is formed. So what is the product of glycolysis? Product of glycolysis is 2 NADH and 2 ATP. Ultimately it will give me 8 ATP, but this 2 NADH in aerobic respiration, it will go to mitochondria and then it will produce. In anaerobic respiration, only 2 ATP. Substrate level ATP, 2 ATP. Glycolysis is also called EMP pathway. The final product of glycolysis is 2 molecule of pyruvate. 2 molecule of pyruvate. Now these pyruvate will undergo link reaction. Pyruvate will undergo link reaction and pyruvate will form acetyl coenzyme A, as shown here. Pyruvate will form acetyl coenzyme A. In that process, NADH is formed. This acetyl coenzyme A will further take part in the Krebs cycle, in which you can see first 6 carbon citric acid is formed, then it lose 1 CO2 and form 5 carbon alpha ketoglutaric acid, then it becomes succinic acid and NADH and ATP is formed, which release FADH and also one NADH is formed. So overall, if we see in one Krebs cycle, 3 NADH and 1 FADH is formed. Along with that, 1 ATP is also formed. Here the GTP form will get converted into ATP through a reaction called coupled reaction, right? So overall, how many ATP will be formed here? 12 ATP from one cycle. For one glucose, there are 2 Krebs cycle, so total 24 ATP, out of which how many is substrate level? 2 ATP is substrate level, 22 ATP is through the oxidative phosphorylation. Now, in this process also NADH and FADH is formed. In Krebs cycle, how many substrate level ATP formed? 2. How many oxidative? 22. Total ATP 24. Right? In link reaction, total ATP formed 6 from 2 molecule of NADH. So total ATP 38. How many substrate level? 4. How many through oxidative phosphorylation? 34. Then finally, all these NADH and FADH, how do they produce the ATP? So now here, this you need to pay attention. ETS, many of the students have some doubt here in ETS. So first of all, where does ETS take place? So it takes place in the inner mitochondrial membrane. How many complex it has? So it has 5 complex. What is the order of complex? So it like 1, then 3, then 2, then 4, and finally 5. What are the name of these complex? So that is given. Remember these names. From name also question is asked. But if I ask you, where does NADH go? Goes to which complex? So NADH goes to complex 1. Where does FADH go? FADH goes to complex 2. Right? Now, if you see, from 1 NADH will go and NADH will oxidize, produce 2 electron, FADH oxidize, produce 2 electron. Both the electron goes to cytochrome B through ubiquinone. Then from cytochrome B it goes to FES. From FES it goes to cytochrome C1. So this is the movement of electron. From cytochrome C it goes to cytochrome C1, it goes to cytochrome C. Cytochrome C is considered as the mobile electron carrier, whereas the ubiquinone is the proton carrier. Ubiquinone is the proton carrier and cytochrome is the mobile electron carrier. Then finally the electron through copper, cytochrome A, A3 and copper, it goes and get accepted by the oxygen. It get accepted by the oxygen. So who is the final electron acceptor? Oxygen is the final electron acceptor. Oxygen is the final electron acceptor. Now, which complex has copper present in it? Complex 4. Which complex is impermeable to H+? Complex 2. Which complex do not have FES? So answer is complex 4 and 5. In which complex the H+ move from intermembrane space to matrix? Complex 5. Which complex do not take part in electron transport system? Complex 5. So in this way question will be asked. Now due to this movement of electron, what will happen? H+ will move from matrix to intermembrane space. H+ is moving from matrix to intermembrane space, from matrix to intermembrane space, right? Now, because of this, H+ concentration will increase here. So high concentration this side, low concentration. So high concentration to low concentration. So ATP will be formed. For each H4H+, 1 ATP is formed. From 1 NADH, 3 ATP is formed. From 1 FADH, 2 ATP is formed. Now here the respiratory pathway, also called as amphibolic pathway, right? Here in this case, fatty acid can enter into the respiratory pathway. Similarly, amino acid can enter into the pathway. Similarly, they can also go out from the pathway, right? So one is catabolism, another is anabolism. So since it is both catabolism and anabolism, so it is called amphibolic pathway. RQ value, it is different. In case of carbohydrate, it is 1. In case of protein, it is 0.9. In case of fat, it is 0.7. In plant growth and development, so here mainly the question is asked from the different, so here important points are growth rate, very important. Here these graphs are important if they are asked. So from graph here, the absolute growth rate and relative growth rate are important. Then in this case, differentiation, dedifferentiation, redifferentiation. Here the plasticity, again example of plasticity, from here question is asked, keep it in mind. Then the different phytohormones and their role, again simple and remember, but it is frequently asked, so you have to remember. So what you do, you write down the name of the phytohormones and then all the function, you quickly put it on a sheet of paper and keep on revising it twice and thrice, you will get it. Then, so this part is done. Then let's move on to the sexual reproduction part. So in sexual reproduction, first is the structure or of the anther. So anther has, the stamen has two parts, anther and filament. The anther has 4 lobes present. Anther has 4 lobes. Now each lobe, anther has 2 lobes in which 4 pollen sacs are present. Now if we see the structure of these pollen sacs, so pollen sac has outermost epidermis, then endodermis, middle layer, innermost tapetum, and then finally the microspore mother cell. This microspore mother cell undergo meiosis and it form microspore. This microspore will develop into male gametophyte. So this microspore, actually here, this microspore is formed in the form of tetrad, which will form microspore. This microspore will develop into a 2-cell stage in which one vegetative cell is there, another generative cell is there. These 2-cell stage then further become 3-cell stage in which the generative cell divide and form 2 male gamete. 3-cell stage is called matured male gametophyte and 2-cell stage is called matured pollen grain. Then these pollen grain have 2 layers, exine and entine. The exine is made up of sporopollenin, entine is made up of pectocellulose. In this exine is highly resistant structure and entine is a flexible structure, so it helps in the pollen tube formation. Further, if we see here, so in case of gynoecium, so this is the ovule. Basically, the unit of gynoecium is called pistil. It has 3 parts: stigma, style and ovary. Ovary, inside the ovary there can be 1 or more than 1 ovule. This is the anatropous ovule, as you can see. It has integument surrounded. Inside the integument is the nucellus, and then from the nucellus cell will develop the megaspore mother cell. This megaspore mother cell will undergo meiosis and form a linear tetrad. Out of the 3, 3 will degenerate, 1 will act as a functional megaspore, which will undergo 3 free nuclear division, and it will give rise to a 7-cell 8-nucleus embryo sac, right? Since it is formed from 1 megaspore, so such embryo sac is called monosporic in nature. Now, so here if we see the structure of embryo sac, this is important. From here question is asked, right? How labeling: antipodal cell, this is the egg apparatus, central cell, polar nuclei, and this is the filiform apparatus. During fertilization, the pollen tube will enter inside the ovule and it will break the synergids, and there the 2 male gamete are released. One of the male gamete will fuse with the egg, second male gamete will fuse with the secondary nuclei, and this is called double fertilization. Double fertilization is a characteristic feature of angiosperm, in which the first one is called syngamy. Syngamy give rise to zygote, which form embryo, embryo develop into seed. Whereas the triple fusion give rise to primary endosperm nucleus, which is present in present in primary endosperm cell, and that will through mitotic division will develop into endosperm. So here if we see, this is how the zygote is formed. Then the zygote will become first proembryo, then it will develop into the globular embryo, heart-shaped embryo, and then finally the matured embryo. During this development, in most of the case, nucellus disappear. But if nucellus do not disappear, it will be called as the perisperm. Example of perisperm is there. So in case of this, so now here if we see, next is, so in case of embryo, there can be monocot and dicot. Here, two cotyledon, dicot, single cotyledon, monocot. In monocot, the single cotyledon is called scutellum, right? That is a scutellum. You can see from here, labeling part is very important. Labeling of monocot and dicot, both can be asked. So keep that in mind. Then here, as I said, the perisperm which is present, and perisperm, black pepper and beet are the example. Now in the seed, if endosperm is present, then it is called albuminous. If endosperm is not present, it is called exalbuminous. Usually monocot are albuminous, dicot are exalbuminous, but here exceptions are there. Castor is a dicot with endosperm, and orchid is a monocot without endosperm. So exceptions are here. Then here apomixis is there. So apomixis means it mimics the sexual reproduction. It is a type of asexual reproduction which mimics the sexual reproduction, is called apomixis, right? In apomixis, the diploid cell will directly develop into embryo without undergoing meiosis, formation of egg or fusion of egg. This will lead to the polyembryony. Apomixis, polyembryony. Now, they are helpful in conserving the hybrid character of a seed. That's why usually hybrid seeds are propagated through apomixis. Now, coming to the next principle of inheritance. So here Mendel has studied 7 characters. He has studied 14 traits of each character. For each trait, he has developed true breeding variety. So there are total 14 true breeding variety. And he has given this 3 law. He has given the law of dominance, law of segregation, law of independent assortment. Law of dominance, law of segregation, law of independent assortment. In law of dominance, what he said, then in a heterozygous condition, one of the allele will express, it is dominant, allele which not express, recessive. In law of segregation, what it said, that in case of heterozygous condition, there is no blending of character. During gamete formation, these allele will segregate. In the law of independent assortment, he said that pair of allele of 2 gene will assort independently during the formation of gamete. That is, if there are 2 allele, let's say capital R, capital Y, cross with small r, small y, and it forms capital R, capital Y, small y, then it will form 4 types of gamete in equal proportion. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. All these 4 are formed in the equal proportion, that is 1/4. This is said by the law of independent assortment. Now, these ratio will change when there is a linkage. So linkage do not follow this rule. Now, so that is why he has given the monohybrid cross. So in monohybrid cross, there is a genotypic ratio of 1:2:1 and phenotypic ratio is 3:1. Phenotypic ratio is 3:1. Now here the formula to find out the number of gamete, 2 to the power n, where n is the number of heterozygote. Number of genotype, so number of genotype you will call 3 to the power n. Number of or type of zygote, so here it will be 4 to the power n, and n is 1 for monohybrid, 2 for dihybrid, 3 for monohybrid, dihybrid and trihybrid, etc. So from here, if question is asked how many gamete, how many genotype, how many phenotype, easily such questions can be done. For test cross, the phenotypic and genotypic ratio all is 1:1. It is for monohybrid. If it is for dihybrid, it will become 1:1:1:1. Now, there is a exception of Mendel's law of dominance, and that is incomplete dominance and codominance. In case of incomplete dominance, if we see incomplete dominance, then here the genotypic ratio and phenotypic ratio both is 1:2:1. Genotypic and phenotypic ratio both is 1:2:1. Here the heterozygous condition will have a separate phenotype which will give pink in color, right? Similarly, here this point is again important, where it says that, let's say there is an allele, let's say capital T is an allele. When it modify, it can have three conditions. It can form normal or less efficient enzyme. Now in this case it will form product, so if product is formed, so it will be same as dominant phenotype. But if there is non-functional enzyme, as it's shown here, when there is non-functional enzyme, so product is not formed, no product, no phenotype. When there is no enzyme, again no product, no phenotype, right? So in the last two cases, when there is non-functional or no enzyme, there is no product, so recessive phenotype. But in case of unmodified or normal or less efficient enzyme, there is a dominant phenotype. Further, if we see codominance, here again the homozygote in genotypic and phenotypic ratio both is 1:2:1. But here example you have to remember, AB blood group is an example of codominance. As you can see here, A and B both are expressed here. Further, here there is a B gene given, and B gene is an example of two things. One is, it is an example of pleiotropy because it is covering two characters. One is the shape of the seed, another is the size of the seed, and also it is showing incomplete dominance. Remember, from here 3 points are there. First is, it is pleiotropy. Then it is showing incomplete dominance, and it is showing dominance. Incomplete dominance in size of the seed, and dominance in case of shape of the seed. Why pleiotropy? Because it is controlling both size and shape, so it is called pleiotropic in nature. Now, when there is inheritance of two genes, so that is called the dihybrid cross. In case of dihybrid cross, the phenotypic ratio is 9:3:3:1. And here these are both dominant, one recessive, one dominant, and here both are recessive. So parental are 9+1. If I say parental, so 10/16. If I say hybrid, this I am talking about phenotype. Hybrid phenotype 6/16, right? Parental is 6/16. So in this way, now if I ask you about the genotype, so let's say This is the genotype. Capital R, small r, capital Y, and tell me what is the probability of this genotype. So here again the formula uses 2 to the power n. So 2 to the power n divided by 16. So it will be 4 by 16. Similarly, capital R, capital R, capital Y, capital Y. So here n value is 0. So 2 to the power n is 0. So 1 by 16. So using this method you can calculate any of the probability. Right? If you have the genotype, then using 2 to the power n formula, you can calculate all the probability. You don't need to remember the genotypic ratio in this case. Now, talking about chromosomal theory of inheritance. So, chromosomal theory of inheritance was given by Sutton and Boveri. Now, here this chromosomal theory of inheritance says that in this case, when they were studying, and when Sutton and Boveri has gone through the Mendel's work, then he realized that Mendel is telling the same thing which I am telling about the chromosome present in pair. The factors are also present in pair. Chromosome segregate, factor segregate. Chromosome assort independently, factor assort independently. So it is same. So he proposed that there is a parallelism between behavior of gene and parallelism between behavior of chromosome. So this is the chromosomal basis of inheritance. Now here this diagram is showing segregation, right? It is showing segregation in which the two chromosomes are segregated, but here it is showing independent assortment which is giving rise to 4 combination, right? So segregation always give rise to 2 combination and independent assortment give rise to 4. Then further this Morgan has studied Drosophila melanogaster and he has given the concept of linkage, and linkage is an exception of law of independent assortment. Now here what was said, that if the distance between 2 genes is small, if the distance between 2 genes is less, as we can see here, then linkage is more. If linkage is more, then parental character is more. See 98.7. So less distance, more linkage, more parental character. More distance, less linkage, less parental character, right? So here basically it says that linkage is inversely proportional to distance, right? Linkage is directly proportional to parental character and linkage is inversely proportional to crossing over. So basically now the recombinant will show the recombination frequency, and using the recombination frequency one can find out the gene mapping, which was given by the student of Morgan, who was Alfred Stewart Went. So he basically said that distance between gene, let's say there are 3 genes A, B and C. So if recombination frequency between A and C is 10%, means they are far. If recombination frequency A and B is 4, then they are comparatively closer, and B and C is 6%. So if this is the recombination frequency, you have to draw the genetic map, then you can simply draw it like this: 10, 4 and 6. So 6 plus 4 is 10. So basically what here, recombination frequency will represent the distance between 2 genes. It represents the distance between 2 genes. Then there are few terms like polygenic inheritance. So polygenic inheritance means what? When more than one gene control one character. So there is, let's say, one character and it is controlled by several genes A, B and C. Then it is called polygenic inheritance. Polygenic inheritance are usually additive. Polygenic inheritance are additive in nature. On the other hand, pleiotropy, when one gene control many character. When one gene control many character, let's say character 1, 2, 3, then it is called pleiotropy. Example of pleiotropy is phenylketonuria is an example, sickle cell anemia is an example, and we have also seen the B gene just a while back. So all these three are example. Now example of polygenic inheritance, skin color in human and height in human. Remember, this polygenic inheritance are influenced by environment. It is also influenced by environment, right? Then, so these are parts of zoology. So let's skip these parts. Then coming to the molecular basis. So in this we will talk about the nucleic acid. So nucleic acid basically is a polynucleotide which is of two types, DNA and RNA, right? So here if we understand, so this nucleotide has three parts. One is the nitrogenous base, another is sugar, and the third one is phosphate. These are the three. Now nucleotide, if you remove phosphate, then it will become nucleoside. There are two categories of nitrogenous base, purine and pyrimidine. Purine as a nitrogenous base consists of adenine and guanine. Pyrimidine is cytosine, uracil and thymine. Purine always pair with pyrimidine, right? Purine always pair with pyrimidine, and it is given by Chargaff's rule. Chargaff's rule says that A plus G is equals to T plus C, or A plus G divided by T plus C is equals to 1, right? This is the Chargaff's rule. Now, according to this question, how it is asked, you will be given adenine, you are asked thymine, cytosine, guanine, etc. So that you can easily find it out. Remember DNA, RNA. RNA will have uracil, DNA will have thymine. No DNA will have uracil, no RNA will have thymine. If you see thymine, just consider it as DNA. This Chargaff's rule is always valid for double-stranded DNA. It is valid for double-stranded DNA. Now, this is the structure. One polynucleotide, another polynucleotide, both are connected with the help of hydrogen bond. Hydrogen bond is present between the nitrogenous base. Two nucleotide are connected to each other with the help of, this is phosphodiester bond. Phosphodiester bond. This is the hydrogen bond. This is the hydrogen bond. Now, DNA, one turn, 360 degree is called one turn. So let's say this is one turn will have 10 base pair. One turn has 10 base pair, and the diameter is 2 nanometer or 20 angstrom. Now here length or the pitch of one turn is 3.4 nanometer. So one base pair is 0.34 nanometer. Now central dogma says that DNA form mRNA, mRNA form protein. That is the linear flow of information. Now DNA, it's very long. DNA is very long. How it is packed inside a small nucleus? Of course, it need packing. So packing is done with the help of nucleosome. Nucleosome, histone, H1 histone, DNA. So remember this, histone is octamer. Octamer means it has 8 subunit, right? Now histone is also positively charged. So it will have basic amino acid. Which basic amino acid? Lysine and arginine. Here the histone which is positively charged and DNA which is negatively charged combine together to form nucleosome. These nucleosome form the beads on string structure which ultimately form the chromatin. Chromatin form chromatin fiber form chromosome. So formation of chromatin need histone protein, but higher level of packing, that is formation from chromatin fiber to chromosome, will require non-histone chromosomal protein also. So formation of chromosome will require both histone and non-histone protein. This chromatin can be euchromatin also, heterochromatin. I have explained earlier in cell only that one is active, another is inactive. Then this historically how DNA evolved. So here Griffith experiment is there, in which he has first of all reported that there is some transforming principle which is converting the R strain, which is non-pathogenic, into pathogenic S strain. But he just said something about transforming principle. He could not explain what exactly is transforming principle, and that was explained by three scientists, Avery, MacLeod and McCarthy, and for that they have used enzyme, DNase, protease and RNase, and they found that whenever I am using DNase, then the transformation is failed, means DNA is responsible for transformation, that's why DNA is the transforming principle. But he has used enzyme. So usually enzyme used reactions are not very acceptable. So it was not accepted by many scientists. Then comes another experiment by Hershey and Chase. And Hershey and Chase has unequivocally proved that DNA is the genetic material. And for that he has coated the protein, coated the DNA with phosphate, P32, and sulfur with S35. Now, when he radio-labeled protein, then there was no radioactivity in E. coli. So, no radioactivity in E. coli means protein did not enter into E. coli. So, protein is not entering. When he radio-labeled DNA, then he observed radioactivity in E. coli. What does that mean? Means DNA is entering into E. coli. So, DNA is the genetic material. So this provide an unequivocal proof that DNA is the genetic material. So this Hershey and Chase experiment has proved that DNA is the genetic material. Then further, the RNA world, in this topic it says that the earlier the earlier genetic material was RNA. And why is it so? Because RNA performs several activity, and during the early time one molecule performing multiple function, it is usually favorable. That's why RNA might be the genetic material, but RNA being unstable because it has ribose protein, and ribose is more reactive as compared to deoxyribose. Hence gradually deoxyribose, that is DNA, replaced RNA as the genetic material, because RNA is less stable, more reactive, and DNA is more stable. So for a storage form of genetic material, DNA is more suitable, right? So that's why early world was RNA as genetic material. Gradually the DNA become the genetic material. Then replication, as Watson and Crick has given the double helical structure of DNA, they have also given the concept that the DNA replication must be semi-conservative, but he hasn't performed any experiment. Then gradually Michelson and Stahl has performed that experiment, and he has had this experiment, and he shown that the DNA replication is semi-conservative. Now for that what he used? He has used heavy isotope, not radioisotope, heavy isotope. N14 is the normal isotope of nitrogen, but he used N15. Now N15 having a greater mass, if both strands are N15, then it will be heavy. If both strands are N14, it will be light. If one strand is N15, one strand is N14, so it will be called as a hybrid strand in terms of mass. So N15 heavy, then hybrid, and then light. So what he did, he first synthesized the DNA which is both N15, so it is heavy. Then he grown the DNA into the light, that is N14. So what he observed, that the DNA he find is of intermediate, that is hybrid DNA he got. Then in the next generation he said that out of 4, 2 is hybrid and 2 is light. In the next generation, out of 8, 2 is hybrid, 6 is light. In the next generation, out of 16, 2 is hybrid, 14 is light, right? So this gave him an appearance that through this he proved that the DNA replication is semi-conservative. Now here how question is asked, you will be asked that tell me after 3 generation what will be the ratio of hybrid to light chain. So see, first generation all hybrid, second generation half hybrid, half light chain, right? 1:1. In the third generation 2 hybrid, 6 light chain, so ratio 1:3. In fourth generation 2 hybrid, 14 light chain, so ratio 2:14, so it will be 1:7, right? So in this way question will be asked. So remember this Michelson and Stahel has used N15 heavy isotope of nitrogen to prove that the DNA replication is semi-conservative in nature. Then next is about the replication. So in replication, it was found that the mechanism of replication, that the DNA both the strand do not separate because it will require a lot of energy. So what happened? Strand open very partially, and in one strand continuous synthesis take place, and in other strand discontinuous synthesis take place. The one which continuous synthesis is called leading strand, another is called lagging strand. Leading and lagging strand. Lagging strand has Okazaki fragment, and these Okazaki fragments are ligated together with the help of DNA ligase. Here here in this way the DNA polymerase will synthesize the DNA in 5' to 3' direction, right? Ultimately, these Okazaki fragments are also joined, and then 2 complete separate DNA is formed. One strand will be new, another will be the old one. Then next is once this replication is done, then in central dogma we have seen another process which is transcription. In transcription what happened? DNA pass on its information in the form of RNA. So here what will happen? Here in transcription it take place in a transcriptional unit which has promoter, structural gene and terminator, right? One of the strand is template, another strand is called coding strand. Now here what will happen? Template strand will form the RNA. So in this case, first of all, the RNA polymerase, which is also called DNA dependent RNA polymerase, will go and bind to the promoter. In this process sigma factor play a role, and then during elongation sigma factor leave and the RNA polymerase elongate, RNA is formed, and finally when the terminator end is there, then the rho factor will come and release the RNA. In this way RNA polymerase is also coming out. So in this way initiation, elongation and termination, the transcription process is completed. Remember here sigma factor and rho factor both play a very important role. Once the translation is done, then the formed RNA is called hnRNA, which undergo processing, in which there is 5' capping, poly A tail of 200 to 300 A, and then there is splicing of intron. You move the non-coding part. Exon is the coding, intron is the non-coding part, so it is removed. Now this processing is not required in prokaryotic. That's why in prokaryotic translation and transcription goes side by side. Now these mRNA which is formed after processing will move into the cytoplasm. These mRNA have certain code which is called genetic code. Genetic code is triplet in nature, which was explained for the first time by George Gamow, then later on by Hargobind Khurana, and then later on further it was Marshall Nirenberg. So all these three scientists' name is important: Gamow, Khurana and Nirenberg. They have said that DNA, this genetic code is triplet in nature. Further, there was another enzyme, Severo-Ochoa enzyme. It synthesized RNA in a template independent manner. Gradually, this is the genetic code and these are the character, simple character are there. Finally, these code will be get translated, and in translation tRNA has an important role. These tRNA will get charged by addition of amino acid. This tRNA will come and bind to the codon part. Anticodon, codon will come together and they will bring amino acid. This amino acid will get transferred. So here in translation there are three steps. First is the initiation. In initiation, what will happen? In the initiation step, first initiator tRNA will come and join to the AUG, which is the start codon, and then next codon, let's say next codon is GAG. So next tRNA will come and join here with amino acid, right? So then gradually when two tRNA are very close to each other, amino acid get transferred from one to other and a peptide bond is formed. In formation of this peptide bond, ribozyme has a very important role to play, and this is also called elongation. In this process, RNA will move from 5' to 3' direction. Ultimately, when the stop codon come, and stop codon is UAG, UAA and UGA. They are also called nonsense codon because they don't have any amino acid or tRNA. So when the stop codon come, then release factor will come and bind to the stop codon and these polypeptide will be released. Now this translation cannot take place without the UTR. UTR is the untranslated region which is present in both 5' end and 3' end. So after this translation, these gene which is expressed can also be regulated, and unit of gene regulation is called operon. So operon, we have to study one important operon, that is lac operon. It is also called inducible operon. Here when the lactose is absent, here the inducer is lactose. When lactose is absent, the I gene produce repressor will bind to operator. When operator bind, then the RNA polymerase access is not there, so no transcription will take place. But when inducer is present, repressor is inactivated, and this lead to formation of beta galactosidase, permease and transacetylase, and then the expression is there. So if lactose is absent, no lac expression. If lactose is present, there is expression of lactose. So this is a kind of negative regulation, although lac operon is also under positive regulation, but we don't have to study that in detail. So remember, in lac operon, repressor bind to operator. What is inducer? Lactose is the inducer, or allolactose is the inducer. I gene will form repressor. This formation is constitutive in nature. And then this is the zoology part, so this part I will skip. Then in the biotechnology part, so basically here what we do, that we take a particular gene and then insert it into a DNA. Now for that, first of all, we will isolate DNA, right? So DNA isolation step is given here. So first there is an isolation of DNA. This is how DNA is isolated. Now in this isolation we use chilled alcohol, right, to precipitate the DNA, and then through spooling we take it out. Other component we digest using lysozyme, cellulose and chitinase. So when we isolate the DNA, then these DNA is cut into small fragments. This is cut into small fragments using restriction endonuclease. Now restriction endonuclease cut a gene at a particular point, and for each restriction endonuclease there is a restriction site. This restriction site usually have a palindromic sequence, as you can see. Remember this sequence, GAATTC, right? So this is called the palindromic sequence from 5 prime to 3 prime. If you read it, it always goes like this. So this is the palindromic sequence. So it cut which bond it cut? It cut the phosphodiester bond on both the strand. So these endonuclease cut the particular strand, we get the desired gene. Now this gene is cloned using PCR, in which there is denaturation, annealing and extension. So through PCR we will do the copying of the inserting gene. Then foreign gene is inserted into the vector. Both vector and the plasmid are cut using the same restriction site. Using the same restriction endonuclease they are cut, and then ligation will give rise to recombinant DNA. Now that ligated fragment can be checked with the help of gel electrophoresis. Here agarose gel is used, and in that the fragments are separated as per their size. Longer the size, slower will they move. So this side is positive, this side is negative. So DNA will move from positive to negative, and the smallest will move fastest and the largest one will move slowest. Here remember, ethidium bromide is used, which will give a orange color when it is exposed to UV light. For that we use a cloning vector. For inserting the plasmid we use a cloning vector, and that cloning vector, this plasmid is the cloning vector. It has several cloning sites, it has origin of replication, it has selectable marker, as you can see, ampicillin, tetracycline. Cloning site, there are total 8 cloning sites. This diagram you have to remember. It is very important diagram. From here a lot of question is asked. How many antibiotic resistant site it has? Ampicillin has which resistant site? How will you distinguish between recombinant, non-recombinant? How will you distinguish between transformant and non-transformant, right? So all are important. Now here, once the DNA is inserted inside, then it is selected using the selectable marker. Selectable marker can be antibiotic resistant marker, or there is another which is called beta galactosidase, which will give colored selection. Means blue means non-recombinant and white means recombinant. So this is also called blue-white screening. It is also called blue-white screening. Other plasmid which are used is called TI plasmid, right? TI plasmid is also very important, and in some cases bacteriophage is also used as a vector. So TI plasmid is another type of vector, bacteriophage is another type of vector. Now once you get the recombinant DNA, you need to insert it into the E. coli. For that you need to make it competent. So competent you can make it using either through the bivalent cation like calcium, or you can use micro injection, you can use gene gun, or you can use disarmed pathogen. So these are some of the ways how you will insert your desired gene into the E. coli. So this is the step of PCR, denaturation, increase the temperature at 92 degrees Celsius, it will denature. Then annealing, you lower the temperature, annealing, 2 primers will join. These primers are DNA primer, and then extension, deoxynucleotide and Taq polymerase are there, so it will cause the extension. In the end you will get 2 double-stranded DNA. If you repeat it to several cycles, you will get millions of copy. For example, 30 cycle is giving 1 billion copy. So denaturation, alleling and extension together they form one cycle. Finally, when these E. coli are there, then they, using the bioreactor, they can be isolated and then they are processed, packaged, and that is called downstream processing. That is called downstream processing. So, these parts are, so here just 2 parts are important, are from the botany, so that part I will cover, and rest are the zoology part. So here if I see, so first of all, this point, formation of genetically modified crops. So here 2 things are there. One is development of Bt cotton. So it is important. Bt cotton, here the gene, so here 3 genes are important, remember, Cry1AC, Cry2AB and Cry1AB. These are the 3 genes which will be synthesizing a particular toxin which will not let the pathogens to grow onto the cotton. Now these are, and what are the pathogens? These are the cotton bollworm and corn borer. So basically they are host specific, right? They will target. So if you want to attack cotton bollworm, you will use, so these 2 genes are there, and Cry1AB for corn borer. The name of the protein is called Cry, and the name of the gene can be Cry1AB, AC, Cry2AB. So here the gene is derived from bacteria Bacillus thuringiensis, and that gene is inserted into the plant using TI plasmid Agrobacterium tumefaciens. Earlier when this was not done, then simply this bacteria was spread onto the plant. But now we insert the gene, and this cotton now can be protected from this cotton bollworm and corn borer. Similarly, RNA interference. In RNA interference, what we do? We target a mRNA using a sequence of RNA, which is called RNAi or interfering RNA. Now here what happened? The mRNA, this RNA will go and bind to the mRNA and form a double-stranded RNA. Now when double-stranded RNA is formed, then there is no transcription. There is no transcription. If there is no transcription, then the protein will not be formed and the organism will die. So what we do here, naturally it is found in 2 cases. It was found, one was in viruses and another in the transposon. But human have done this. So there is one example given of nematode and plant. So what we did, we targeted the mRNA of a nematode, and as per the mRNA we have created a DNA. This DNA it was inserted into the plant. Now plant synthesized the RNA, and this RNA when taken by the nematode die. So in this way we are silencing the gene. So this is also a method of gene regulation which silence the expression of gene in particular organism. So one of the gene regulation we have seen in Lacoperon, another we have seen in the here in the translation. So I think this is it. So now what you need to all to do, that read, revise, and once you have revised each chapter, then you can again go through this video. Next time you can increase the speed because all things are covered here, and if you have already covered, then you can skip and go to that part of video which you want to understand, right? So that is it for today, and in coming sessions we will cover the PYQs and if needed some specific topic related to the exam. So thank you all of you for being with me for so long. Thank you.